生命起源于何处？又将归于哪里？记录、察觉、唤醒，小小的细胞中隐藏着未知的秘密。当生命科学成为人的延伸，我们看见彼此更细微、更光亮。西湖大学生命科学学院致力于研究生命现象、疾病机理，探索发现生物医药的新技术和疾病诊治的新方法。在这里，我们是无畏的探路者，从萌芽一个想法，到开始一次试验，大胆猜想，小心求证，一场场冒险，一遍遍反思，不断追问生命现象的本质。在这里，我们是进取的创新者，平等包容，学科交叉。无论是讲席教授，还是青年科学家、学生、行政人员，我们彼此尊重，自由思辨。在这里，我们是梦想的守护者，鼓励大家去提问，去想象，保持对科学最纯粹的兴趣与好奇。探索未知的科研世界，每一次思考都是对生命科学边界的求索，每一次验证都是对人类生命健康的守护。敬畏、探寻、发现，我们将共同创造更加美好的未来。
尊敬的各位来宾，本次会议即将开始，请您尽快就座，并将手机等通讯设备关闭或置于静音状态。谢谢您的合作。
Ladies and gentlemen, the event will begin shortly. Can we please ask for you to kindly take your seats? We would like to remind you again to switch off all mobile phones and put electronic devices to silent mode. Thank you for your cooperation.
Okay, we'll get started. Good morning, dear speakers, colleagues, and trainees. Welcome to the 2021 We Academic Forum. The theme of the forum is infection and immunity. So the stated purpose of the forum is actually to celebrate the new opening of our new campus, Yungu campus. But as expected, everything is behind schedule, so we're not ready to hold the forum in the new campus. We have to hold it here. But I think the spirit is correct, so we are really happy on this occasion that our new campus will be soon ready for use. So I think speaking as a non-immunologist, I can speak for many of you in the audience that immunology is the hardest subject to learn. In fact, when I was looking for faculty positions in the late 90s, if you look at the ads for faculty position for immunology, oftentimes the deadlines were extended because of lack of qualified applicants. I think the recent years, the success of cancer immunology has really brought a renaissance back to immunology. Now it's one of the hottest areas in research. And also with the current ongoing COVID-19 pandemic, it really reminded us there's a lot of gaps in our knowledge in immunology and infection. So I think this forum is very timely. So of course, the most important part of the meeting is the lineup of speakers. So the organizing committee, I think, except myself, everybody else has done a lot of work. Dang Shengli, Chang Xing, Xu Heping, He Danyang, Liu Zixu, and Zhou Ting are the responsible parties who bring together this outstanding lineup of speakers. We'll have a combination of online and in-person presentations. So they cover a wide range of topics from plant immunology to the interface of neural and immune systems, and of course to cancer immunology and also SARS-CoV-2 mechanisms. So I think this meeting is also sponsored by the Westlake University and the Westlake Laboratory of Life Sciences, and also the World Young Scientists Summit as part of their meeting series, and also is sponsored by Xihu Miwei. I think it's Lucy Micro. It's a startup company funded by our own Tao Liang. So let's thank Liang for supporting this meeting. And also finally, I'd like to thank our administrative team led by Kaina. I think they were testing, and also our team from the outside to really run this meeting both online and offline. They were testing the setups till I think 3 o'clock in the morning. So let's give them a round of applause for their dedication. Finally, I want to say to the meeting, the main purpose of the meeting is, of course, for the trainees. So I hope the trainees will get inspirations from our successful speakers and scientists and find your own interest and niche, and eventually you'll be the future in life sciences that will help hopefully bring the pandemic to the end and also solve major problems in life sciences. I wish I hope the meeting will be a resounding success, and thank you very much for coming. Oh, I now turn the podium over to Zhou Ting, who will chair the first session.
Good morning, everyone. My name is Ting Zhou, and I am a faculty member of Westlake University School of Life Science. So I will be the session host for the morning first session. So we're very happy to have Dr. Akiko Iwasaki as our first opening keynote speaker. Akiko is a professor of Yale University Department of Immunobiology and also Department of Molecular Cellular Developmental Biology. Her lab studies innate and adaptive immune response to a variety of viruses and how this virus infection leads to disease consequence. So her lab made a lot of contributions to this field, including the TLR7, TLR9 signaling in tissue-specific dendritic cells, and the prime and pool vaccine strategy, as well as endogenous map in human genome. So for these early accomplishments, she was recognized as Howard Hughes Medical Investigator since 2014. She was also elected to National Academy of Science in 2018, National Academy of Medicine in 2019, American Academy of Microbiology in 2020, and American Academy of Arts and Science in 2021. And more recently, since the start of this pandemic, as a virologist and also immunologist, Akiko and her lab quickly turned to be a major task force working at the forefront of COVID-19 research. Together with the clinical team, her lab published a lot of important papers addressing key questions in SARS-CoV-2 study. I believe her talk will cover part of that. And outside the lab, she also very active in the social media like Twitter. You can follow her Twitter, Virus Immunity. And there she will be very patiently to explain and communicating COVID-19 facts and also her scientific findings with the public. So she's also quoted by a lot of TV reporters and newspapers as for her expertise insights. And she's also considered one of the top 50 experts to be trusted during this pandemic. And also not only as a good scientist, she's also very great and supporting mentor for the trainees. She's also well known for her advocacy for the women and underrepresented minority groups in the science and medicine field. In today's talk, Akiko, I think she will talk about the immune response to SARS-CoV-2. With that, let's welcome Akiko to give her talk.
Thank you. Thank you very much. Can you hear me? Yes? Okay, I'm going to share my screen. All right, so today, as Ting mentioned, I would like to talk about our recent work on immune responses to SARS-CoV-2. One of the key insights that the immunology community is getting is the sheer variation in COVID-19 disease outcomes. This has to do with, of course, the host, such as the age of the host, as well as the sex, as well as pregnancy status and other conditions that might accompany a person's immune response and infection. And we, together with a number of collaborators, have delve into the immune responses to SARS-CoV-2, looking at patients that are in our hospital with moderate, severe to lethal COVID, and dissecting the different types of immune responses that happen. And our insights include, for instance, the severe COVID patients having larger amount of viral titer, and that are having misfiring of the immune responses that correlate with the immunopathology that we see in COVID patients. We also demonstrated that the timing of the immune response is important, that the neutralizing antibodies that develop early in the immune response is helpful, while those that develop later are not beneficial. We've also done a lot of work on looking at autoimmune responses that happen during COVID, as well as the neuro-COVID and the impact of saliva viral load on disease outcomes. We also turned our attention to looking at differences in sex and immunity. And this in particular, we focused on early phase of immune responses in the patients that are hospitalized. And we found, for instance, that early phase immune response, particularly T cell activation, is elevated in female patients, while those in the male patients were reduced, and that correlated with the worsening of the outcomes. And we've also looked at the impact of COVID and pregnancy, and have reported one severe case of infection that resulted in infection of the placental cells, as well as respiratory tract infection resulting in impact in the placenta. So today I'd like to basically talk about three separate studies. One study has to do with how different mutations that accumulate in the variants impact immunity induced by the vaccines. The second story is a mouse model of COVID-19 that enables us to dissect the different effectors involved in clearance of the pathogen. And finally, I'd like to talk about our ongoing work on long COVID pathogenesis. The first study I'd like to share with you was led by Dr. Carolina Lucas. She is a very talented postdoctoral fellow in the lab. Together with Dr. Chantal Vogels, she is a brilliant postdoc in Nathan Grubel's lab who does viral surveillance. And Dr. Insi Yildirim, who is a pediatric infectious doctor who collaborated with us on the story that I'm about to tell you, along with a number of collaborators. So the study design was to enroll healthcare workers who had either been previously infected, that are indicated in the blue, and previously uninfected, in the purple people. And what we did was we collected blood at baseline prior to vaccination and 7 days and 28 days post first dose of mRNA vaccine, and 7, 28 days or 70 days post second dose of the mRNA vaccine, and compared their antibody and T cell responses. What we showed is that the mRNA vaccines are generally very good at inducing high levels of antibody. So on the top is the anti-spike IgG. The middle panels are the S1 domain of the spike, and the bottom is anti-RBD, the receptor binding domain of the spike. And you can see that at baseline, if you look at the right panel, the vaccinated, previously infected people had some levels of IgG to begin with. But after one dose of mRNA vaccine, which is the time point 1, you start to see already a huge elevation in their antibody titers. And then they maintain very high levels of antibody. Whereas previously uninfected people also get there with 2 doses of mRNA vaccine. Of course, they start out with no antibody to begin with, but this quickly catches up to those who have been previously infected and vaccinated. And when we examine the ability of the mRNA vaccines to elicit neutralizing antibodies, so that this is using an authentic virus of the original SARS-CoV-2 lineage, what we see is that, again, if you look on the right, the patients, the participants who were previously infected are indicated by blue dots, and those who are not are indicated by purple dots. And you see that there is some baseline levels of neutralizing antibodies in the previously infected participants, but this levels goes really high, even just after 1 dose of mRNA vaccine, and maintained very highly. Whereas those participants who had not been exposed to the virus are able to catch up after the second dose. So this is all good, that the mRNA vaccines are eliciting robust neutralizing antibodies. We also demonstrated that the T cell immunity is robustly induced by the 2 doses of mRNA vaccines. This is looking at 28 days post second dose of mRNA vaccine. On the top are the CD4 T cells, and on the bottom are the CD8 T cells. And shaded in purple are the previously uninfected individuals, and shaded in blue are the previously infected. And what I've done is to just sort of highlight the activated T cells, which are HLA-DR positive and CD38 positive. And those types of T cells are observed very readily when we stimulate the T cells with the spike protein from the original lineage A, but also when we tested against a P1 variant of concern. We also saw a very robust CD4 and CD8 T cell response. There was some reduction in the CD8 T cell response to the spike of the P1 variant. However, this definitely remains above background. Next, we turned our attention to the different types of variants that are circulating or that have circulated in our community. And on the panel A on the left, what I'm showing you is the 16 different isolates that Carolina was able to purify out of the nasopharyngeal swabs of people who are infected in the community and genotyped by Chantal. And what you see is that there are these gradients of variants that have different amino acid mutations that accumulate in the spike region, all within the N-terminal domain, the RBD, the RBM, and the furin cleavage site. And on the right, what I'm indicating is the neutralization titer from these various individuals that were vaccinated twice with mRNA vaccines. So against the ancestral strain, I told you that vast majority of people, except for 2 people, develop high levels of neutralizing antibodies. And if you go down the list of variants, it really doesn't make a big difference until you get to about the Delta variants in the greens. You start to see a significant drop in the neutralizing antibody capacity induced by the vaccines. And this coincides with the mutations such as the L452R mutation, which single-handedly has the most impact on escape from neutralization antibody, which is shown in the right. And you go down this list even further, you see the epsilon and eta and kappa, you start to see some more decline in the neutralization titer. But when you get down to the gamma and the beta variants, they're the most evasive or have the highest resistance against the neutralizing antibodies. And on the right, what I'm showing you is the impact of each amino acid mutations within the spike region that have the capacity to escape this neutralization. What's rather striking about the finding is that if you look at the inter-individual variation in the neutralizing antibody response against the variants that are listed across the screen here, you see that people who develop very high levels of neutralizing antibody against the ancestral variant strain have the sort of a capacity to have cross-reactive antibody responses all the way across to the beta variant. Whereas people who started out with lower levels, they tend to drop off in their ability to neutralize variants much quicker. And of course, the 2 people that I mentioned somehow were unable to develop neutralizing antibodies, even though they had quite a good levels of ELISA detectable anti-spike and RBD antibodies. So if you separate these participants into those who are vaccinated on the bottom, they're all vaccinated, those who are previously infected on the bottom versus previously uninfected on the top, what you see is that those who had never been exposed to the virus but have been immunized twice with mRNA vaccine are able to control, of course, the ancestral variant strain, but their ability to neutralize different variants demonstrates rapid decline in their ability to do so with variants that are indicated here. Whereas those who had prior infection and then were vaccinated are able to maintain cross-reactive antibodies, across many, many different variants. So this isn't a big surprise because prior infection does confer another exposure to the antigen. So it's almost like having a priming dose prior to getting the vaccines. So this study demonstrated that the mRNA vaccines do elicit robust antibody, neutralizing antibodies and T cell response against the spike. And previously infected people are able to do so even better than previously unexposed individuals. And that there are distinct mutations in the variant that confer different degrees of escape from the neutralizing antibodies. And certain mutations do appear to synergize to escape the neutralizing titer, such as the Delta H69, V70 combined with the E484K mutation. And finally, we believe that these data sort of illustrates the continuous need to conduct viral genome surveillance across the globe so that we can anticipate what type of variants might be arising that may dominate the different population. Currently, the Delta is dominating the world. However, there may be new variants that arise in the future that might require variant-specific vaccines. So now that we've learned much about these vaccine-induced neutralizing antibodies, we wanted to understand other components of adaptive immunity in conferring resistance against the virus. And this was enabled by a strategy that Dr. Ben Israelo a infectious disease fellow in the lab, developed in the very beginning of the pandemic, which is to develop a flexible mouse model of COVID-19. This question really was elicited by a number of different studies looking at the correlate of protection against SARS-CoV-2 infection. On the left is a Nature Medicine systematic review of different vaccines or infection that generates different amounts of neutralizing antibodies that have a very nice predictive ability to look for protective efficacy. And it's not only the neutralizing antibodies that are important for protection, the non-neutralizing viral binding antibody can also engage the Fc receptor on immune cells to control the virus. And finally, T cells are obviously important for generating antibody responses, as well as acting as effector cells to control infected cells. So there are some really nice evidence out there from real world data that look at these correlations, but we wanted to understand what are the relative importance of these effector functions. So Ben took the approach first to look at the role of B and T cells in clearance of the primary SARS-CoV-2 infection. To do this, he utilized a model that he created, which is to transduce mice of any genetic background with AAV encoding human ACE2. And this enables any mouse of any genetic background to become susceptible to SARS-CoV-2, any human isolates that we have in the lab. And then he can measure disease and viral load in different tissues. So on the bottom of what I'm indicating is the infectious viral particle per mil of within the lung after infection of the wild type animals that are transduced with human ACE2. And you see that there's a very rapid and complete clearance of the virus within 7 days. However, if you look at the MUMT mice in which there are no B cells, these mice are also able to completely clear the virus within 14 days. And at 7 days, there are some low levels of virus detectable in these animals, indicating that B cells are not necessary to control primary infection. However, when Ben took rag knockout animals, these are mice without any T or B cells, these animals were unable to control the primary infection even at 14 days post-infection, and they actually developed chronic infection with SARS-CoV-2. This illustrated the necessity of T and B cells, the adaptive immunity, for controlling primary infection. He then turned his attention to the importance of T cell subsets, CD4s and CD8s, in controlling primary infection. To do this, he again took advantage of the human ACE2 transduction system. And on the top, what he did was he transduced wild-type animals with human ACE2. And then he depleted either CD8, CD4, or CD8 and CD4 T cells and measured viral titer in the lung at 14 days post-infection. And what he saw is that depletion of CD4s or CD8s alone had no impact on the clearance of the virus, indicating that these subsets are not required for clearance. However, when he depleted both of these subsets, then he started to see some animals still harboring virus at 14 days, indicating that you need T cells for complete clearance of the virus. It turns out that in these animals that are depleted of CD8s and CD4s, there was also reduction in antibody responses because of the necessity of CD4 T cells to elicit, provide help for B cell to produce antibodies. And on the bottom, he did the same exact experiment in the MUMT animals that don't have any B cells. And in this setting, what he saw was that depletion of CD8s or CD4s had basically dramatic impact on the ability of these mice to control the virus, indicating that in the absence of antibodies, CD4s and CD8 T cells play a major role in controlling the virus. And when he depleted both CD8s and CD4s, he converted these mu-MT mice into a RAG knockout animal setting in which there are no adaptive immunity present to control the virus. So what about the role of B cells and T cells in protection against reinfection or vaccine-mediated protection? To get at the reinfection question, first Ben used a adaptive transfer system to see if antibodies or T cells are sufficient to confer protection against infection. To do this, Ben took serum, which contains antibodies, or T cells from these animals that were already immune to the SARS-CoV-2 virus and transferred them separately into RAG knockout mice that are transduced with AAV encoding human ACE2. He challenged these animals with SARS-CoV-2 and measured virus in 7 days. And what he found is that the T cell transferred animals were able to significantly reduce the viral load in the lung within 7 days. And some of them completely clear the virus. So indicating T cells are partially responsible for clearance of the virus. However, the serum transferred animals, none of them had any detectable virus within 7 days, indicating the ability of the serum antibody to completely control virus infection. Then Ben took advantage of the mRNA vaccine to create a situation in which animals have varying degrees of antibodies. To do this, Ben took either 1 microgram, 0.3, 0.1, 0.03, 0.01 microgram of mRNA vaccine, gave it to a K18 mice. These are animals that are transgenic for human ACE2. And then he measured antiviral antibodies in these animals. There was this really beautiful dose-dependent response of antibodies generated by different doses of mRNA vaccine, 1 microgram being the best in the blue, followed by 0.3 in the red, 0.1, and so on. And then at 0.01 microgram, there was no detectable antibodies. He took these animals, he challenged them with SARS-CoV-2 and monitored for disease by weight loss or survival. And what he saw was that the mice that received either 1 or 0.3 microgram of mRNA vaccine were completely protected from disease or death, whereas animals that received 0.1 microgram, there were about half of the animals died, and there was disease as observed by weight loss. And any animals getting less than that amount, 0.03 or 0.01, or the mock immunized animals, they all succumb to infection within a week or so. So this gave Ben a really great platform to calculate the correlate of protection against disease or death. And what he found is that there's a beautiful correlation between the amount of antibody against the S1, the pre-infection stage on the x-axis versus the weight loss at 6 days post-infection on the y-axis. So the more antibody animals generated, the less weight they lost. So based on this calculation, he was able to calculate the amount of IgG required for death and survival, as well as weight loss. And so there's a very nice correlation between the amount of antibody developed, either neutralizing antibodies or anti-spike antibody, and the protection from disease and death. Finally, Ben wanted to understand the role of CD8 T cells in protection against reinfection or mRNA vaccine mediated protection. So he created 2 cohorts of animals, one that have been fully vaccinated with the mRNA vaccine on the top, and the one that has been recovered from prior infection, a convalescent animal. And then he challenged these animals with either the Washington strain of the SARS-CoV-2 or the beta variant. And before doing that, Ben also depleted CD8 T cells from some of these animals. And ultimately, what he found was that all animals that received the vaccine or prior infection were completely protected from death, while all the animals that were not immunized succumbed to infection within 4 days of infection. And when he looked at the viral load, infectious viral titer in the lung of these animals, vaccine and convalescent animals had no detectable infectious titer in 2 days post-infection, whether they were depleted of CD8 T cells or not, and whether they were challenged with the original strain or the beta variant. So this really demonstrated the power of the mRNA vaccine that relies on antibodies, and that the CD8 T cells are dispensable from protection against SARS-CoV-2 infection. So this study illustrated that T and B cells both clear primary infection, that in the absence of one, the other can compensate. However, if you're lacking both T and B cells, animals are unable to control primary infection and they remain chronically infected. Adaptive transfer studies demonstrated the complete protection from serum antibody and partial protection conferred by T cells. And in convalescent animals or in vaccinated animals, CD8 T cells were dispensable for protection against a challenge by ancestral or the beta variant. And finally, he determined the levels of antibodies needed to provide protection based on the mRNA vaccines. So this study illustrates the sort of power and the benefit of the mRNA vaccine in conferring some level of mucosal immunity. So even though mRNA vaccines weren't designed to generate mucosal immunity, we now know from various studies that we can find mucosal antibodies of the IgG and IgA isotypes in the saliva and the nasopharynx of mRNA vaccinated individuals that can have neutralizing activity. That would prevent infection altogether. And even if the virus is able to infect and replicate within the host, there are antibodies and T cell based mechanisms that prevent the spread of that virus. And we already know there's a very effective ability of these mRNA vaccines to prevent symptomatic infections, which then leads to transmission enhancing activities such as sneezing and coughing. And finally, we believe that even transmission may be further prevented by coating of the virus with antibodies generated by the vaccines. So at all levels, vaccines are inducing some level of mucosal immunity due to some crossover from the systemic to the mucosal tissues. However, we believe that the future vaccines, the next generation of vaccines, could benefit from having a mucosal targeting approaches. That's something that we are working on right now. And just finally, I'd like to describe what we are doing right now about the long COVID and why we're interested in this question. So long COVID is happening in millions of people around the globe. That is affecting multiple organ systems that are listed here. These are just some of the over 200 different symptoms that are associated with long COVID. And this is striking in that a respiratory tract viral infection resulting in incredible organ system involvement, including systemic impact, neuropsychiatric, head, eye, ears, nose, and throat, endocrine, cardiovascular, pulmonary, musculoskeletal, and GI tract symptoms. And how can a respiratory virus infection result in involvement of so many organs? Well, one of the hypotheses that we are thinking about is potential establishment of viral reservoir. Now, this can be either infectious viral particle or remnants of virus or just RNA or protein of the virus that's lodged somewhere in the tissue that's not accessible to the nasopharyngeal swabs. And this could trigger chronic inflammation. Another hypothesis we are pursuing is that the COVID infection could trigger autoreactive lymphocytes that target different organs, such as those targeting the antibodies targeting different organs, tissues, and cell types, as well as T cells. There are other hypotheses out there that can explain long COVID, including the tissue damage, dysbiosis of microbiome, as well as latent viral reactivation that's often seen in long COVID patients. So there's evidence for both the viral reservoir and autoantibodies for long COVID. The viral reservoirs have been seen in COVID patients months after the initial infection. This study found, for instance, there are some nucleocapsid staining in the enterocytes of patients that had COVID months before. This is 5 out of 14 patients had positive staining, and 3 out of 14 patients had PCR positive for viral RNA after 3 months in the intestine. There's also evidence of autoantibodies that develop in COVID-19. This is during the acute phase of COVID-19. We collaborated with Aaron Ring, who is a professor in our department where Ting used to work. And Eric Wang and Yili Dai are trainees from his lab. Tiangyang Mao and John Klein are students in my lab. They collaborated to look for autoantibodies in acute COVID patients. This was enabled by Aaron's new technology called rapid extracellular antigen profiling, which enables comprehensive and high throughput screening for autoantibodies based on yeast display library, where human exoproteome are expressed by these yeast cells. And you simply have to incubate the patient's antibody with these yeast cells, and you pull it down using magnetic beads of the yeast that are bound by the IgG. And then you can decode the antigen using deep sequencing. And this enabled us to detect numerous autoantibodies that are found in COVID patients against both immune cells, immune factors, cytokines, interferons, as well as autoantibodies against target tissues. So this is just demonstrating some of the different tissues and different autoantigens that are indicated in the in different rows. And each patient is a column. And patients are divided into severe, moderate, mild, or asymptomatic. And the brighter the yellow color, the more autoantibody there is against that particular autoantigen. So you can see that most patients have multiple autoreactivity against different antigens. There is no unique autoantigen that's found across these patients. However, there are some common ones that are found. And the striking thing is that they are antigens expressed in the brain, vasculature, connective tissue, cardiac, hepatic tissue, saliva, skin, GI tract, and many others. So this illustrates the possibility that COVID-19 can lead to autoantibody induction, which then targets different tissues in different antigens within different tissues to trigger pathological outcome. And there are other reports demonstrating autoantibodies in both acute and long COVID, including the G protein coupled receptors that are already known to elicit a lot of the pathologies. So currently we are working with a number of teams to understand immunological and physiological determinants of disease pathogenesis in long COVID. And this is the only way to inform rational diagnosis and therapy for the millions that are suffering from the consequences. And this was generously supported by HHMI. So I want to end by thanking and acknowledging the authors of the 3 studies that I was able to describe to you today. And I already highlighted the first authors of each paper. And just want to end by thanking the amazing lab members that I have the privilege of working with on a daily basis to try to figure out COVID-19 immunity and to try to dissect long COVID so that we can eventually help these patients in in the near future. So thank you for your attention.
All right, thank you, Akiko, for covering so many important topics in the COVID-19 patient immune response. Now we have like 7 minutes for the questions. Okay, Dr. Yang Xinfu.
Hey, Akiko. I have a question related to that two patients that failed to respond to have a neutralized antibody. I wonder whether it's because they lack of a T cell response to RBD, or is lack of B cell response to RBD. Do you have any data for that?
Yeah, so what's intriguing about these two individuals is that they don't have any, as far as we can tell, they don't have any immunodeficiencies. They do develop ELISA detectable anti-spike and anti-RBD IgG, and yet they don't have the capacity to neutralize the virus. So I think the virus neutralization assay that we're using is using the authentic variants, and it's quite stringent. You know, the fact that they can't neutralize it, functionally these anti-RBD antibodies are not being able to block the virus from infecting the cells. So we haven't cloned these BCRs and try to start dissecting the mechanism, but they do develop ELISA antibodies, and as far as we can tell, they also develop some levels of T cell response. So there's something intrinsically faulty about their the particular BCRs that they're using.
And another question about mRNA vaccine. You mentioned about IgA. Traditionally, when you do intramuscular injection, IgA is very, very low. But I'm not sure when you mentioned about mRNA vaccine can induce IgA, it's because mRNA vaccine have some unique feature, or the IgA still is very low?
Yes, absolutely. So if you compare it to a convalescent individuals, the IgA level is quite low. You can detect it though, and it doesn't happen with adenoviral vector vaccines. So there's something about the mRNA vaccines that's eliciting low levels of mucosal IgA. And there's some evidence that some of them may even be secretory IgA because they can detect secretory components. But again, the level is much lower than a convalescent person. So this level of sort of crosstalk is there, it's present, but it's not efficient.
Yeah. Dr. Bin Su.
Hello, Kiko. Hi, this is Bing.
Hi, Bing.
How are you? I got a question about this long COVID. You showed that there is viruses in the gut. I just curious, in that situation, the host has any immune response, like antibody or T cell responses to those viruses in the intestine?
Yeah, so I only picked that one figure out of that paper, but the whole paper was about the maturation of antibody response that happened in COVID-infected individuals. And essentially, yeah, these people who have these intestinal, PCR positive or antigen positive biopsies, they also are maturing antibody responses over time. So I think what this means is that even in patients who don't have GI tract symptoms or long COVID, there may be a little bit of depot of antigen that happen in the intestine that may be helping them to develop even more somatically mutated and mature antibody responses. However, we also think there's a flip side to it, which is that in some patients, that is actually acting as a source of chronic inflammation if they're not able to clear, the right amount of pathogen there. So we're still, looking at these two possibilities, and intestine is one of the potential target of reservoir.
Thanks.
Thank you.
Okay, Dr.
Hi, Kiko. Beautiful talk. This is Isaac. I was actually wondering about the last part about what are your thoughts about the effects of COVID on the brain. I know you've done some work in that area, and do you think the autoantibodies that you found with Aaron are involved, or is it direct viral-induced, injury to the CNS that could be part of long COVID?
Yeah, thank you, Isaac. So there are papers, including ours, that detected infection in the brain, and I think it does happen, especially in some severe cases. We don't currently know if that happens in long COVID patients, and it's a little bit invasive to probe that. However, there are studies, including one from Eric Song and Shelly Farhadian, who looked at the CSF from patients with neurological COVID. And they did find autoantibodies as well as antiviral antibodies in the CSF, suggesting that there may be either local infection and or autoantibody induction within that CNS, and that may be leading to some of the symptoms. So, we haven't ruled in either of these things. I think infection in the brain can happen and does happen. How frequently does that happen in the long COVID is a question we don't currently understand.
Okay, Dr. Hai.
Thank you.
Hi. Hi, Akiko. I'm intrigued by the autoantibodies. You think that's special to the long COVID compared to acute and resolving infection? And what about other type of viral infection? I thought when there is inflammation and viral infection typically are associated with some levels of autoantibodies. So could you comment on the spectrum of antigen or the degree of antibody production? But that's where the specificity is.
Yes, so that's a really important question. REAP was recently developed, so Aaron is actually going backwards, trying to understand whether infection other than COVID might induce similar kinds of autoantibodies. So he's collaborating with Charles Dela Cruz, who's a pulmonologist at Yale, who has an archive sample from flu infection prior to COVID. So that's something that they're going to figure out pretty soon. I suspect that you might see some of these autoantibodies develop. And your first question about whether autoantibody may particularly associate with long COVID, that's something we're actually directly looking at, because we have these patients that we characterized from March, April of 2020, who had these autoantibodies. And now we are surveying these patients that are obviously discharged from the hospital. And the survivors, we are now collecting samples and information from these people to see how many of them actually develop long COVID. And the sad thing is that people with that kind of severe disease, they often suffer from post-ICU syndromes, as well as, upwards of 70-80% of these people still have symptoms way past 6 months of hospital just discharge. So I suspect that a lot of them do have long COVID. And if any autoantibodies are associated with that, that's a critical question that John Klein and others are currently asking.
Due to the time limit, let's stop here. Thank you, Akiko, for this insightful talk. Thank you.
Thank you so much. Thank you.
All right.
Thank you.
Our second speaker in the first session is Dr. Isaac Cho. Isaac is associate professor in the Department of Immunology at Harvard Medical School. He received his undergrad training in biochemistry at Harvard College, and he got his PhD in immunology in Harvard Medical School under the mentorship of Dr. Michael Carroll, working on immune responses in neurodegeneration. He then trained with Dr. Tom Manaitis' lab as a postdoc fellow on ALS. He also did his second postdoc fellowship on the neurobiology of pain at Boston Children's Hospital under Dr. Clifford Wood. He studied his own lab in Harvard Medical School in 2014. So his lab studied very interesting topic focused on interactions between the nervous system, immune system, and the microbe in the host defense and inflammation. His work has found that the nociceptor neurons directly sense bacteria and immune mediators to produce pain. And also, recent work has also shown the immune modulators also regulate itching during the allergic inflammation. And Dr. Chu has received a lot of rewards, including NIH Director's New Innovator Award, Chan Zuckerberg Initiate Ben Barres Award, and Burroughs Wellcome Fund Investigator in the Pathogenesis of Infection Disease Award. And I think his topic today is nociceptor sensory neurons, regulation of the barrier immunity. Okay, welcome, Dr. Isaac Chu.
Thank you so much, Ting, and it's a pleasure to be here with Westlake University opening. It's an honor to be invited. So I'm going to talk to you today about neurons and immunity, and the topic is how do these neurons called nociceptors regulate barrier host defense? So when we think about the nervous system, we have to realize that every organ and peripheral organ in the body, as well as especially the barrier tissues, such as the gut, the skin, the lungs, and I'll talk to you today about the meninges as well, are densely innervated by sensory and autonomic nerves. And the role of these nerves are to take in stimuli from the environment and also to regulate barrier functions. So our lab is really interested in how these nerves crosstalk with microbes as well as with immune cells in these key barriers. So today I'll tell you basically two stories, one on the gut and one on the meninges, which are two key barrier sites. Okay, so the neurons we study are those that mediate pain. Why are we interested in pain? Because actually pain is one of the 4 cardinal signs of inflammation. So it's been linked since 2,000 years ago by Celsus to, in a sense, to the immune response. But it's always been thought of for a long time as a symptom, but now we're realizing that the pain-mediating nerves are actively participating in regulating inflammation. And the neurons that mediate pain are called nociceptors. So this term nociceptor was coined by Charles Sherrington to describe the pain reflex circuit, where he hypothesized there must be mechanisms that the body uses to sense noxious stimuli, and then this is transduced into an unpleasant pain sensation and a withdrawal reflex. Well, the neurons that mediate pain, the nociceptors, reside within the dorsal root ganglia. They take in signals from peripheral barrier tissues and transduce them up to the spinal cord, where this is then processed to the brain as a pain signal. And these neurons are able to detect many different types of noxious stimuli, and their role is to protect us from danger. So these include noxious heat or cold, tissue injury, force, reactive chemicals, as well as potentially microbes and pathogens. And on the one hand, behaviorally, this leads to a withdrawal reflex to protect us from further exposure to these danger. But also these neurons can do something else at the molecular level. So one of the things that we found is that these nerves, like immune cells, can directly sense pathogens, just like the immune system. So this is just an illustration of multiple molecular mechanisms by which nociceptors sense microbes. They're able to detect pore-forming molecules from Strep pyogenes and Staph aureus, such as streptolysin S and hemolysins, and this induces pain production. Also, gram-negative pathogens can activate nociceptors through LPS or flagellin via TLR4 and TLR5, as well as TRP channels like TRPA1 and TRPV1. And also, beyond bacteria, Candida has also been found to directly activate nociceptors to reduce pain. So in a sense, these nerves can act like sentinels for pathogen invasion. And then as a consequence, they also signal locally to the immune system. So what's called an axon reflex, where an action potential is transduced along neighboring branches of the nerve fiber that leads to release of neurotransmitters and neuropeptides that then also signal to the immune system or the vasculature. So these neurons are known to immediately induce vasodilation, permeability, as well as signal to the immune system to modulate chemotaxis and priming. One of the key ways that this occurs is through neuropeptides. So neuropeptides, including substance P, CGRP, and VIP, are stored in the peripheral terminals of these nociceptors. And when calcium comes in, they are released, and then these can act on immune cells that have receptors, cognate receptors for neuropeptides, such as mast cells, which can detect substance P via this GPCR called MRGB2. This induces a very significant mast cell activation, so this nerve mast cell axis. Also, CGRP is a neuropeptide that I'll get into that can act on multiple types of immune cells, including dendritic cells to induce IL-23. And also VIP, another neuropeptide, can act on ILC2s. So this is a growing list of interactions between neuropeptides and immune cells that is of interest. And in the big picture, then these nociceptor immune interactions via neuropeptides and other mechanisms are key for barrier immunity. So this is a series of papers that have been shown from our lab and others that neurons in the skin as well as in the respiratory tract regulate the outcome of infection, as well as autoimmunity and asthma. This is just one of the studies we had published recently showing that in skin infections, TRPV1 expressing nociceptors release CGRP, which then suppresses neutrophil recruitment, and that this facilitates survival of a bacteria called Strep pyogenes. And we can block that by having antagonists against CGRP and a release of CGRP that can improve the outcome of infection. So I'm going to tell you today about the gut and the meninges, which are two key barrier sites of our body and how nociceptors regulate them. So the first part is on Nikki Lai's work, who's a graduate student who was working on how nociceptors regulate intestinal host defense against Salmonella. So Salmonella is a major gram-negative bacterial pathogen that causes 1/4 of all diarrheal disease. And it invades the small intestine of the gut, transiently colonizing the ileum, and then invading via Peyer's patches, which are specialized lymphoid organs in the small intestine ileum. And they invade via these M cells, which are specialized epithelial cells. Upon translocation, then macrophages take them out of the gut and they spread systemically. So we wanted to ask, do neurons play a role in host defense against this bug? And to study nociceptors, we used different ion channels to target them. So one of the key ones we use to target nociceptors is TRPV1, which is a large pore cation channel that has been shown to detect capsaicin, which is the active ingredient in chili peppers, and heat, noxious heat, as well as low pH. TRPV1 is expressed in the large proportion of gut innervating nociceptors. And just of note, it recently received recognition that David Julius, who discovered TRPV1, won the Nobel Prize this year in physiology and medicine. So how do we target these neurons? Well, we use a TRPV1 DTR mouse, so TRPV1 expressing human diphtheria toxin receptor, and then we injected these mice with DT to kill off TRPV1 expressing neurons, and then infected these orally with Salmonella. And what you could see here is that these mice lacking TRPV1 fibers did significantly worse in Salmonella infection. So they had more bacterial load in the ileum Peyer's patch in the early time points of infection compared to vehicle-treated controls, as well as spread to the liver by day 5 post-infection. As a second parallel strategy to target nociceptors, we used another ion channel, sodium channel NaV1.8, which is important for action potential generation in nociceptors, and it's not expressed in non-neuronal cells. So here we have a NaV1.8 Cree that we crossed with a ROSA26 DTA mouse. So this will ablate NaV1.8 lineage nociceptors from development, and Cree negative mice are wild type controls. So we infected these orally with Salmonella and then looked at bacterial load at day 1 and day 5 in the small intestine and liver. And what you could see is that this phenocopies the TRPV1 neuron ablated mice, where significantly more bacteria was recovered from the ileum and Peyer's patch at day 1 and also spread to the liver by day 5. So these neuron deficient mice are deficient in host defense. And turns out these neurons, the TRPV1 lineage neurons, which reside in the dorsal root ganglia, can directly sense salmonella. So here, what we're doing is growing dorsal root ganglia neurons in culture and stimulating them with the bacteria or with vehicle control. And you could see that subsets of these neurons were directly activated by the bacteria or products in the bacteria. And they also respond to capsaicin, so indicating they express TRPV1. So the neurons are able to sense the bacteria. And I'm summarizing a lot of data. It turns out that the way that these nerves then signal to the gut is through these Peyer's patch M cells. And M cells are these key epithelial cells that sample antigens from the lumen of the gut, but are also exploited by the salmonella for entry. So they reside on top of the follicles of the Peyer's patch, and they have these characteristic microfold shapes. That's why they're called M cells. And so the nerves are actually directly juxtaposed to these M cells underneath the dome of the Peyer's patch. Here's an image of this. You can see these mature M cells marked with the protein GP2. And Salmonella utilizes GP2 as its binding protein on the M cells, and then they translocate through the M cells. So what we found is that these nerves actually regulate the homeostasis of the M cells. So when you target nociceptors, either by using NaV1.8 DTA or TRPV1 DTR, this leads to increased numbers of M cells. So remember, the epithelium is always turning over. The neurons suppress the differentiation and turnover of the M cells. So you can see at baseline, there are more M cells per follicle in the absence of nociceptors. And what we found was that the way that the regulation worked was through this neuropeptide called CGRP. So CGRP actually suppressed the numbers of M cells along the dome of the Peyer's patch. This in turn was also quite important, not only from preventing entry of bacteria which use M cells, but also by suppressing M cells, it allowed a better microbiome homeostasis. So SFB, which are key bacteria that induce Th17 cells, but also protect against enteric pathogens, are able to attach to enterocytes, but not well to M cells. So by decreasing the number of M cells, there's also more SFB, which then protects against salmonella. So overall, this shows that nociceptors in the gut maintain the homeostasis of the epithelial layer as well as the microbiome to protect against the pathogen. Okay, so this is in the GI tract. And I'm going to tell you some unpublished work that led by two very talented postdocs in the lab, Felipe Ribeiro and Li-Wen Deng. And this is in the meninges. So the meninges I consider also a key barrier site because it is basically the barrier of the brain. And I think you might hear some of more talk in this meeting from Dr. Yoni Kipnis, who is one of the leaders in understanding meningeal immunity. The meninges is made up of three layers, the pia, arachnoid, and dura mater, and it basically wraps around the brain, and it's key for protecting the brain from invasion as well as from trauma. The outermost layer, the dura, is actually quite different than the brain itself in that it lacks a blood-brain barrier, and it actually has fenestrated blood vessels as well as lymphatic vessels, which has been shown by Yoni's lab to drain antigen from the brain, as well as populated densely with the immune cells. So it's a very interesting barrier site for immunity, but also something that maybe people don't appreciate as well is it's also densely innervated by pain fibers. So people in the pain field know this well because these nerves are what thought to mediate headache. So these are some images we took and with two different strains of mice in the lab with a NAV-CRE DD tomato, which will label nociceptors that are NaV1.8 positive, as well as Calc-A GFP DTR, which labels CGRP expressing nerves. And you can see this is in the dura. So this beautiful innervation pattern of the dura by pain fibers or nociceptive neurons. So also, like I mentioned, the meninges is densely populated by immune cells. So in the brain itself, there's a lot of microglia, but in the dura, there's a whole set of, both myeloid as well as adaptive immune cells and lymphocytes. So you can see these are just some recent studies that looked at the dynamics of immune cells at these border sites. And so actually Yoni's lab and also Marco Colonna's lab recently found that the skull bone marrow also circulates cells back and forth from the bone marrow to the meninges. So it's a really interesting dynamic area full of immune cells. And our question is, do nerves play a role in regulating the immune response here? And specifically in the context of bacterial meningitis. So as the term implies, it is bacterial invasion of the meninges, which affects a large proportion of people a year. And it mainly occurs by spread from the hematogenous route, so from the blood via to the meninges, and then from the meninges into the brain. And mortality can still be quite high. So bacterial meningitis, even if you survive it, there's often post-infectious neurological sequelae. So that includes cognitive issues as well as chronic headache and hearing loss. So understanding how meningitis occurs and also how the host responds against meningitis is quite important. So in order to study meningitis in the lab, we set up two mouse models where we used two different bugs, group B strep or Strep agalactiae, which is the leading cause of neonatal meningitis in humans, as well as Strep pneumoniae, which is one of the leading causes of adult meningitis in the elderly, as well as in children. So what we do is we infect mice IV with different doses of the Strep agalactiae or Strep pneumoniae. At a high dose, this will cause, you can see sequential invasion of the dura first, which is outermost layer, and arachnoid, and then the pia and chorioplexus, followed by the brain. So we can study this is bacterial load recovery from each of these tissues. We can study whether nociceptors are involved in this infection. So what we did is we used the NAV 1.8 Cre DTA mice, which have targeted ablation of nociceptive lineage neurons, and the Cre positive mice lack nociceptors, and the Cre negative mice have nociceptors. And then we did infection, in this case with Strep pneumoniae. And interestingly, we actually found decreased bacterial load in the dura as well as in the brain, but no effect on the bacteremia in the blood. So nociceptor absence actually protects against bacterial invasion. And this was accompanied by significantly more influx of neutrophils and monocytes, which are important in clearance of the bugs. I do want to point out that one of the key mediators of nociception in the meninges is CGRP. So this neuropeptide I mentioned earlier, it's released into the meningeal, into the dura, where it's thought to act on the vasculature as well as potentially on immune cells in headache and migraine. So there's a lot of drugs that target CGRP right now to treat migraine. So we wanted to ask, does CGRP also play a role in meningitis? So in order to ask that, we used CGRP knockout mice. So CGRP has two isoforms, calc A and calc B. Calc A is the major one expressed by nociceptors. And what you see here is infection with calc A knockouts versus wild type or hets, but there's significantly less bacteria than is able to invade the arachnoid or the brain in the CGRP deficient mice. By contrast, if we inject the mice systemically with CGRP in a dose that phenocopies migraine models, this actually potentiates invasion. So you get worse invasion of bacteria into the dura, subdural, chorioplexus, and brain, but no effect on bacteremia. So CGRP is negatively regulating the outcome of infection. And we also found that CGRP is released during meningitis by nociceptors. So what we could do, what Felipe did here was he took a skull meninges explant at 24 hours post-infection versus uninfected mice, and he could detect more CGRP there. And this is a time course of that. And then if we remove nociceptors using NAV 1.8 Cre DTA mice, this CGRP level was significantly less. So nociceptors are releasing this neuropeptide. And also we could find bacteria sometimes juxtaposed to CGRP expressing nerves in the dura. So this is imaging of the bacteria in red and CGRP fibers in green. So could the bacteria be activating these nerves to produce CGRP? So that's what we asked here. So Felipe took trigeminal ganglion neurons, which are the ones that innervate the meninges, cultured them, and then added strep pneumoniae to these nerves. And what you could see is in calcium imaging, a subset of nerves that it responded directly to the bacteria as well as the capsaicin, and also that in vitro this induces CGRP release. So we think one of the mechanisms that this is happening is through a pore-forming toxin called pneumolysin. So the strep pneumoniae uses this to facilitate invasion, but also we think it could activate nerves, because here when we use a strep pneumoniae strain that lacked pneumolysin, there was less neuronal responses to the strep pneumoniae. Okay, so the CGRP is being released by nerves. What is it doing? So in order to understand how it's regulating the immune response, we did single-cell RNA-seq of the meninges before and after meningitis, which actually hasn't really been done in the bacterial context. So as you can see here, looking at all immune cells, CD45 positive cells, there's dramatic changes in many immune populations before and after meningitis, particularly in myeloid cells like macrophages and neutrophils. And so in particular, we found that macrophages, here labeled in blue by CX3CR1, are closely juxtaposed to meningeal CGRP nerves. And we could see them in this image. And also many of these macrophages, which also express CD206, increase and respond during meningitis. So could the CGRP be regulating macrophages? So in order to study that, we wanted to first see where's the receptor for CGRP expressed. So the CGRP signals via a co-receptor formed by RAMP1 and calc-RL, which is a G protein coupled receptor, and this induces cyclic AMP signaling in immune cells. So looking across the single cell data set, we could see that calc-RL and RAMP1, but actually not so much these other neuropeptide receptors, is highly expressed actually in many, many immune cell types, including macrophages and neutrophils in the meninges before and after infection. And to target it, what we did is we first crossed the RAMP1 floxed animal with the lysozyme Cre. So this allows elimination of RAMP1 in most myeloid cells, including neutrophils, monocytes, and macrophages. And upon infection of these mice, what you can see is lack of RAMP1, the receptor for CGRP, improved the outcome of infection. So there's less bacteria in the dura and the brain, but not effect on the bacteremia. But lysozyme does not specifically target macrophages. It's also in neutrophils. So in order to go after macrophages only, we looked for a specific macrophage marker. So looking at the single cell data set and also looking at some published papers, one of them, PF4, is actually expressed in macrophages at pretty high levels, but not in microglia in the brain. So this is actually a PF4 Cre tomato mouse. So you can see that these meningeal macrophages are labeled and they're lining the blood vessels. So we crossed the PF4 Cre with the RAMP1 flox mouse, and this allowed us to specifically ablate RAMP1 in macrophages. And this also showed significantly worse, so a better improvement of infection with less bacterial load at 24 hours in the brain and dura. So we think macrophage signaling of CGRP is important. And so how does this occur? So what Felipe did was he injected CGRP into mice and did single-cell RNA-seq of mice with and without CGRP after meningitis and compared the immune response. And what he found was that in macrophages in particular, there's a big polarization of the immune response where CGRP would turn on many genes on one side, but also turn off some potentially key genes such as chemokines. And so we think the chemokines such as, here CCL2 and CCL3, which we looked at in vitro, are decreased by CGRP. And also TNF-alpha, which is of course a key pro-inflammatory cytokine that could be involved in host defense. It did not seem to affect macrophage killing of the bacteria. So this suggests that macrophages are being dysregulated by CGRP. And this is kind of confirmed here where in the RAMP1 knockout mice, we also see basically that, you get more monocytes and neutrophils, which we think are recruited by the macrophages via their chemokines and cytokines. So this also begs the question, if RAMP1 or CGRP blockade is so effective, potentially this could be used as a treatment for meningitis. And since there are RAMP1 treatments right now for migraine, we tried using that in our model. So here we use a RAMP1 antagonist called BIBN. And what you can see here is that treating mice with the BIBN during infection can significantly decrease bacterial invasion of the dura and the brain for both Strep Acalactia and Strep pneumoniae. So what I've shown you here in the second part of my talk is that in the meninges, another key barrier site, these nociceptors are impacting the immune response. So actually, the trigeminal neurons are responding to the presence of Strep pneumoniae or Strep Acalacti to secrete CGRP. CGRP, on the one hand, probably drives significant pain and headache, which is a characteristic of meningitis, but also signals to resident macrophages there, which then regulates or polarizes the macrophage response, which then inhibits antimicrobial defenses. And this may be exploited by the bacteria. For example, the bacteria can activate the neurons via pneumolysin. So if we block this pathway, we can enhance immunity against the bacteria. So in summary, what I've shown you is that at two key barrier sites, nociceptors and immune or nociceptor epithelial interactions mediate antimicrobial host defenses. In the gut, TRPV1 neurons regulate the homeostasis of Peyer's patch M cells, and these are key entry points of salmonella, so this protects against salmonella infection. And in the meninges, the nociceptor signal via CGRP RAMP1 axis to macrophages in streptococcal bacterial meningitis. So really, these are just two cases, but we think that understanding how neurons crosstalk with immune cells at these barriers could lead to new treatments for infection and inflammation. And this was all work done by very talented people in the lab. I mentioned Nikki Lai, who did the work on salmonella, and Felipe and Li Wen, who did the work on meningitis. And these are some really great people, members of my lab. And we have key collaborators, including Mina Rao, Jun Hu, Christophe, and Diane on the gut work, as well as Kelly Duran, Dan Levy, Rick Malley on the meningitis work. So, and very generous funding sources. Happy to take questions.
Thank you, Isaac, for this wonderful talk on how these nociceptor neuroimmune interaction can do both antimicrobial, also do protection role in the meninges. Now we have 5 minutes for the question. Yeah, Dang Yang. Oh, Dan Shen, you can go first.
Hi. A couple of questions. First, this nociceptor regulation of immune function. Is it neuronal autonomous function, or do you think it involves circuitry, neuronal circuitry?
It's a good question. So I think it will, depending on the situation. So the work that I've shown is really autonomous. So it's happening locally because of the neuropeptide release. But these neurons will also induce central circuits like stress signaling, which could, have major, feedback loops via the brain to the immune system, such through the HPA axis, for example. So I think that, like we had found actually that pain induces stress, which then actually feeds back. This is in collaboration with Yachi Xu on inducing stem cell loss in the hair follicles. So there's I think there's going to be long distance circuitry induced immune responses and also local nociceptor immune responses.
Okay. The second question, that in the case of a meningeal infection, the CGRP released by the nerve actually inhibits antibacterial defense. So why do we want to do that, and what's the function of that mechanism? Right.
It's a great question. It's puzzling. So I think that possibly it's protective in other cases, like when you have too much inflammation, right? And potentially in traumatic brain injury, or maybe even other pathogen infections where the immune response, too much of an immune response is actually a bad thing. So I think it may depend on the situation. And we know actually in meningitis is a complicated case where actually you have to treat with steroids as well as antibiotics in patients. So, the immune regulation at that border is a complex area, and I think nerves need to tune that. So I hope that answers the question.
Yeah, Danyang.
Hey, Isaac, a very excellent talk. I have several questions. So I wonder if the innervation in the meninges have some functions and even hemostatic condition, like regulating lymphatic drainage or like attracting immune cells from the skull or other regulatory functions?
Yeah, that's a very good question. So as I mentioned, the main role of those nociceptors is to mediate pain and headache. So I guess the question is, when do you have a headache? Maybe when you're feeling sick, but also it helps you want to lie down and sleep, and maybe that helps with the lymphatic drainage point. But like your point is, are there other roles of these in maintaining the homeostasis of the meninges? So that includes potentially regulating lymphatic function and vascular function, as well as the immune homeostasis at that border. So I think that's probably going to be happening.
Thanks. I have a question related to the first part. So there are CGRP positive neurons in the gut as well, intrinsic innervation from the enteric nervous system, right? I just wonder if this CGRP neurons also contribute to bacterial infection in the GI tract.
Yeah, that's a great question. So what you're referring to, there's of course the enteric nervous system, and there are sensory neurons there that express CGRP. We kind of ruled them out in a way because most of those express CGRP beta. And when we looked at CGRP beta knockout mice, there was no effect on the infection, whereas CGRP alpha, which is mostly in the extrinsic nerves, play the key role. But I think that I think we have to think about, the gut is probably the most complex in terms of sensory innervation. So, the different parts of the gut, the small intestine versus the colon, there could be different roles of intrinsic versus extrinsic sensory neurons, and the role of CGRP source from each of these could play different roles in each situation, different depending on the pathogen, the type of inflammation.
Thank you.
Okay, Zixu.
Yeah, I mean, it's a wonderful talk. I mean, I like it, talk very well. So I have like a really quick question. So you show like depletion of the TRPV1 and also Levi-18. So you show the M cells were increased. So I was wondering if this really happened during the Salmonella infection. And the second thing is if this apply to the other like bacterial pathogens, since a lot of bacterial pathogens they use M cells to get into the gut, right? Thank you.
Right. So actually, a lot of you're right. Actually, a lot of the data I showed was actually at homeostasis. So meaning that this is happening even before infection. But we also saw after infection the numbers. So M cells are actually increased in number by Salmonella. So they will induce M cell differentiation. So nociceptor deficient might have even more above the increase that you see. So I think it's regulating when you have homeostasis. But I think that the mice will often be exposed to things that can activate these nerves. So it could be something in the diet, like I mentioned capsaicin. It could be something in the microbiome. So we're thinking the microbiome actually regulates nociceptors at small levels at homeostasis. So kind of what activates the nerves at homeostasis will maintain some low levels of regulation. And then when you have a major damage, which could be a pathogen or it could be something noxious in the gut, then you have a much higher level of regulation.
Yeah, Dr. Haiqi.
Hi, Zach. I just have a quick question. I want to clarify whether I misunderstood. You mean the TRPV1 neurons directly sense the bacteria-derived product in the gut lumen, then interact with the M cells to effectuate the increased number. Is that correct, or?
I think it's theoretical, but I think that the nerves can be can be activated via multiple pathways in vivo. So in vitro, we know it can respond to Salmonella, but whether that's the actual trigger in vivo, we don't know for sure. So. It's right. So I think that's fair, very fair. So I think, enteroendocrine cells are really key in the gut. And those are, in normal situation when there's no barrier compromise, they're the first ones sensing luminal contents. And they actually have some interesting nociceptive characteristics. They have TRPA1, which is another nociceptor ion channel, and they have receptors for short chain fatty acids. So they could be the ones first talking to these nerves via serotonin. But I think, I guess our argument is during barrier compromise, then the microbiome or, salmonella and things can directly talk to the neurons. And I think generally the nociceptors are just tuned to anything that's very tissue damaging. So it could be heat, it could be, noxious things from bacteria, it could be, I guess, also spices which mimic those things. So, yeah.
Liang, for the last question.
Hello, Isaac. Yeah, nice to see you. And I have two questions. The first question is, as you mentioned, the bacteria like Streptococcus pneumoniae activate neurons by agents like pneumolysine. I wonder whether this interaction is specific to the nociceptor expressed in neurons or sensory neurons. This is the first question.
That's a great question. I'll have to ask Philippe. So this is some of the data is a little bit preliminary, so we're still analyzing it. What we found with other pore-forming toxins like alpha-hemolysin or the strep-derived streptolysin S is at low doses, it seems to be specific for nociceptors. But when you get higher to the point where it's very lytic, then every neuron will respond. So it's not, so there might be something about the nociceptor membrane that's a little bit more sensitive to low doses of the toxins. But yeah, once it gets higher, it's just no longer so specific. But I also feel like it's kind of these things where these are noxious, highly damaging molecules. So it may make sense that a pain fiber, would be able to respond, right? When you need it to respond.
Question is rather quick. So, is there any common ways that bacteria can actually fight back to block the neuron sensing mechanism that can benefit to their infections?
Yeah, no, that's a great question. So we have we're very interested in this. So there have been some analgesic bacterial products that have been identified. So like certain probiotics, people are trying to find things that block pain. So there was a lipoprotein called E. coli that was found to block pain. There are some bugs that may block pain to help it, spread. So we actually have a study coming out on anthrax toxin, which we found blocks pain. And we know it's actually causing painless lesions. So maybe in the skin, this will facilitate its spread, from person to person. This is a fascinating area. So bacteria may exploit nociceptors also to activate them. Like there was a nice Cell paper from Mike Shiloh's lab showing that tuberculosis activates nociceptors to induce cough. So that could help it spread. So the whole dynamic of how bacteria target neurons is very interesting.
I think time is up. Thank you, Isaac, for this wonderful talk. And
Thank you.
Okay, our first session in the morning now ends. Now we'll have a tea break. I want to highlight that I would like to encourage all the trainees to ask questions. This talk is a good training opportunity for you to engage with these international speakers or so. So I would like you to bravely ask a question. All questions are welcome. Yeah. I hope you guys can have more questions later. All right. Thank you.
Hello everyone. Welcome to the second session of this meeting. In this section, we will have three outstanding speakers focusing on different aspects of human immunology. So please allow me to firstly introduce our first speaker, Dr. Ren Sun, who currently is a professor in the University of Hong Kong. Before joining the University of Hong Kong, Dr. Sun was a distinguished professor of pharmacology of the School of Medicine, as well as bioengineering of the School of Engineering at UCLA. Dr. Sun's research really is trying to apply systems biology approaches to address critical viral and host interactions. So very recently, his lab has established a platform to profile antibody response at a single amino acid resolution, as well as at a very large genomic scale, which I think he will talk about today. So let's warmly welcome Professor Sun for his talk.
Hello? Hello, can you hear me? Hello? Okay. So first of all, thanks organizer for offering this opportunity to share my experience. Today I will describe the new technology platform established at Hong Kong. It's a transition for me to be from biologist to try to learn immunology. So in the past 30 years, I have been working on virus and looking at how virus and leaving US and move back to China, give me opportunity and also out of necessity to start a new research direction to not overlap with the research has been done in the US. So we are starting looking at how the immune system interacting with the pathogen. And so I will share the experience, how we move from being. So first couple of minutes, I'll describe our biology experience and how does that lead us to immunology. Several years ago, we started to have a systematic exam of the virus genome, and then we mutate every new one mutation at a time across the genome. And we pulled our mutants together and look at the phenotype of all variants. So here, and the length of the bar reflects how well the virus replicates in different conditions. So this is a functional genetic map of the virus genome. And from that functional map at the different conditions, this on this side is the virus replication. Okay. On this side is how virus replicates without adding interferon, and on the right side, my right side, and look at how virus replicates in the presence of interferon. So at the different selection conditions, how each mutant behave, and then the difference between those two conditions reflects how virus interacting with the immune system, with the interferon. And from this, when they mutated, reduced virus replication in the presence of interferon, but did not reduce the virus replication in the absence of interferon. And we combine 8 mutants together, and each mutant reduce replication, and the replication was reduced as a vaccine. So the virus cannot replicate in normal mice, and the virus replication
So we are encountering some technical problem to bring Shenzhen back. So then we will directly move to the next speaker, and then we will call Professor Sun when he is available. So sorry about that. So our next speaker is Dr. Hai Qi, so who currently is a tenured professor of immunology at Tsinghua University. He's also serving as the Dean of School of Medicine at Tsinghua. So Dr. Qi is really one of the international leading scientists in the field of germ center B cell selection and humoral immunology. So original work published from his lab in roughly the last 10 years has really been helped us to understand how high affinity antibodies will generate at a molecular, cellular, and systems levels. So his work has been recognized by many significant international and national awards. So for trainers, I want to mention that Dr. Qi's talk is always very insightful and beautiful. So I hope you will enjoy it. So let's welcome Dr. Qi.
Thanks for that nice introduction. That's put the extra pressure on me. So as Heping mentioned, that part thanks to his graduate work that we have been studying terminal centers for a while, and we try to understand how these remarkable dynamic structure organized and function to produce the vaccine response that we have heard in the first session. And also, the potentially dangerous response from the autoimmune response. Okay, yeah. So the title is two parts. It's a selection and output, but then I decided that I will focus on the selection. But the reason that we study the selection is really because of the, why is it, I don't want to, okay, let me control that. I can control that. So this is the place where our immune response to generate, it's just a pointer, it doesn't work. Oh, here, okay. From here that you output this after selection, memory B cells or plasma cells that underlie our useful immune response, but sometimes when it goes wrong, it can also lead to autoimmunity and lymphoma. The reason behind that is because in order to generate the high affinity antibody, the B cells here need to hypermutate. They have to generate a secondary diversity of their BCR, and that's being selected based on their binding affinity to antigens through a process that's still incompletely understood. And that's what I'm going to tell you about today. So we know B cells produce the antibody, but B cell doesn't just become good plasma cells on their own. It needs to talk to many other cells, and one of the most important one is the helper T cells, that in a nutshell, the B cells need to acquire antigen based on their binding affinity, then present it to the peptide MHC to the T cells that recognize that through their T cell receptor. And T cell will give the B cells a variety of helper signals. Among them, one of the most important is CD40 ligand, that really, it's one of TNF family cytokines that membrane bound, but tell the B cell to survive or proliferate and do a variety of things that is necessary to become a good plasma cell. So this information exchange, of course, is contact dependent and require time. So the two cells have to come together and stay together for a reasonable amount of time in order for the information to transmit. But if you look at the dynamic structure here, the red cells is the germ center cells and germ center B cells, and the blue and the green cells are the T cells. They don't necessarily stay together for a very long time, even though that I have accelerated the movies for 300 times. On average, they actually stay together for 3 to 5 minutes. So it's hard to imagine that it would finish this information exchange within such a short period of time. So in essence, the B cell need to sequentially talk to different T cells. And so that brings an interesting thought experiment. How do the B cells decide to do what they are supposed to do after the selection? So I will list it here. So even though we don't know the molecular details, one can assume the B cell need to have a signaling integrator inside the, be it the epigenetic, be it the signaling pathways, something that need to account for how much help signal they receive from the T cells. And based on the experiment that from many different groups, we know that the B cell will die if it doesn't get any T cell help inside the germinal center. Then we call that a selection failure. But hopefully before they die, they receive some T cell help that I will use an arrowhead as a thought experiment as an indicator. If it receives, then I will rescue the B cell from the death by neglect. But if it continues to receive multiple delivery of help, then hopefully they reach accumulate the signal to a certain point that it will pass a selection threshold and become successfully positively selected. Then the B cells can go on to do, either stay inside the germinal center to amplify the response or go out to become memory cells or plasma cells. And that decision point, how did the B cell make that decision, is really the topic of entire different talk that is still being actively pursued by many different labs in the world. Today, I'll focus on this question is, before that, what if we miss one of, so that the B cell do not get a frequent T cell help, then what happens is the B cell would presumably will oscillate within this two threshold is until either die or they're being positively selected. So the essence of being positive or successfully selected, it seems to be, given the dynamic nature of the germinal center, is that the B cells need to productively interact with the multiple T cells at a sufficient frequency. If we can sort of translate have this scientific question. Now, how that is being managed, you can imagine in this dynamic context of dynamic interaction, everything can happen in random. So some B cells will just randomly bump into different helper T cells in a productive way, then they will acquire sufficient help to be positively selected. That still can work. But we wonder whether that can be facilitated in any way. And I think that one of the interesting features of the immune system, when we get to the level in the single cell dynamics, it's the stochasticity and the deterministic, it's always a two side that one has to consider. And if there is something that can facilitate the process, it typically can always, you can find evidence for that. And that's what we are looking for. We want to ask whether T cells really can tell good B cells, even from a distance. That would make a contact-dependent B cell help more efficient. If that's the case, then we assume that there should be a process that for B cell to attract the T cells according to their affinity. So the point is you want to select the high affinity B cells, and whether naturally rising high affinity B cell would have a better ability to attract the T cell help, then they can focus on to themselves to receive multiple T cell help within reasonable period of time to be selected. But with this sort of idea, I think for the audience, the students sitting in the audience, you probably scratch their head. Do you want to really go on this potentially goose hunt to find evidence for that? And it's actually not easy to convince students to do that. And I wasn't, at the beginning, wasn't brave enough to look for the evidence until something that showed up actually by sort of an accident. So when we were studying a transcription factor BCL6 in the T cells. So I think for some of you, you will be very familiar. This is considered a master regulator for helper T cell inside the germinal center. If you knock it out, then you lose the germinal center T cells and you lose the germinal center itself. But when we have a heterozygous BCL6 mutant in the T cells, the TFH cells or the helper T cell inside GC look normal, but they show a very obvious defect in terms of their calcium signaling beneath the TCR stimulation. And when we were imaging these cells, we realized an interesting phenomenon. I will show you this movie where the B cell is green and the knockout cells or heterozygous knockout is green, red, and the wild type is blue. And we will watch how these two cells chasing the green B cell. This is B cells will highlight. Whenever there's a T cell bump into it, it will highlight with a white line. So what you realize is that this wild type T cell seems to be able to bump into the T cells, B cells more frequent, or the other way around is the B cell seems to attract the wild type T cells better than the heterozygous knockout can sense that B cells. So at the time we realized that this dynamic feature suggests something that controlled by BCL6 is impacting on how T cell perceive the B cells. And eventually we realized BCL6 at least in part control the calcium signaling inside the T cell, be it triggered by the TCR or be it triggered by GPCR. So, but that gave us a more concrete evidence, say, this almost certainly is a chemo-attracted process. It's something that's different between these two types of cells in a calcium-dependent way that's sensing the B cell. But then there has to be a chemo-attracted receptor and ligand involved. And that gave us a more concrete hypothesis to look for. And so a former graduate student, Liu Bo, when he did rotation in the lab, he started this heroic effort that we start to screen for chemokines expressed by germ center B cell. And the experiment that we set up, now looking back, it's really important to use the CD40 stimulation, but I will explain toward the end. So basically, germ center B cells, in vivo, we briefly stimulate with agonistic CD40 antibody to mimic the T cell help for a very short period of time, then look for all the chemokines that potentially be expressed, and we realized that there's one chemokine, CCL22, that is dramatically upregulated by more than 1,000-fold. And we know CCL22 act on CCR4 receptor that has known been expressed by the T cell. That gave us potentially a ligand and receptor pair that would regulate the germ center B cell attraction of T cells. And so that's our hypothesis to test. So of course we want to verify the T cell express CCR4 receptor, so we made a CCR4 knockout as a negative control. So helper T cells express CCR4, regulatory cells as previously have been found that express high level of CCR4. The regulatory T cells inside the follicle, we call the TFR cells, also express CCR4 similar to TFH. And this is just a quantitation saying that T cells do express, and as expected, a conventional Treg, for lack of no better words, a conventional Treg, express the highest level. And functionally, the Treg cells are attracted to the chemokine better than everybody else. But within the follicular context, the TFH cells respond to CCL22. It has a cousin chemokine working on the same receptor called CCL17. So we can here. Then the TFR cells do not respond functionally to the chemokine. So that creates a situation inside the follicle. The chemokine potentially would only attract or more strongly the helper T cell. We don't understand fully why regulatory cells do not respond chemokine when they express the chemokine receptor, but like many other things, the Treg cells have the apparatus, but they respond differently than the conventional T cells. So that's an interesting topic, but that's aside. So with this chemokine receptor expression, now we want to understand inside the germ center whether there's really evidence for B cell to attract the T cells using this receptor. So because we had the previous finding with the BCL6 heterozygous knockout, then we set up exactly the same system, but now we are comparing the T cells that lack a CCR4 receptor or express the receptor. And the whole system is, they have antigen specificity, OT2 cells recognize OVA, and the MD4 cells recognize HEL, and we can use a model antigen, HEL-OVA, to generate the germ center response. What you will see is a focus on particular B cells, and we'll see how these B cells be chased or attracting the two type of T cells. Whenever there is a contact, we'll highlight with the broken white line. and it will, hopefully you can appreciate how the red B cells would differentially attract these two type of T cells. So you can see the cyan colors T cells bump into the red cells more frequent than the green knockout. Of course, because in order to do this experiment, we always have to do the fluorescent protein transgenic. So it's important to actually verify what we saw isn't because we are using two different fluorescent protein transgenic line. So here is the wild-type comparison using the same CFP-GFP transgenic. Now, if you look at this red cells, you can see there are blue and the green cells bump onto this red cells more in a comparable manner. So of course we have to quantitate because in all the experiment we have wild-type internal control, and we know the tissue volume we image, we know the number of cells inside the tissue volume, and so we can have a very well controlled quantitation of how frequent they hit each other. And there's a clear, when the T cells lack CCR4, they have a much markedly reduced ability to chase the B cells. And we verified, just to make sure this is not a knockout developmental defect, we also conduct experiment with shRNA knockdown of the mature T cells, and we got recapitulated the same finding that suggests well, rule out the possibility it's a developmentally different in terms of the T cells. And then we verified the CCL22-17 double knockout B cells will have the same phenomenon, show the same phenomenon of differentially attracting T cells, and their contact durations within our imaging period, it's always comparable, meaning that what we are affecting is really the frequency or how easy the B cell attract the T cells. So with that, of course, we would expect that the German center is not being maintained as well because the T cells are visiting or because of lack of the receptor or the ligand, they don't visit the B cells as frequently, and the B cells do not receive sufficient help. So there's no repercussion in terms of T cell expansion or follicular helper cells abundance, but there's clear repercussion for the German center size, and that can be recapitulated. Either we knock out the receptor from the T cells or the ligand from the B cells. Well, I think the slide is this is just, well, actually I want to took out this slide, but well, it's basically not, well, I thought I didn't show. The knockout cells, knockout B cells and wild-type B cells comparison, it's become a busier movie, is that you watch the blue cells and green cells, whether they how they attract the T cells, and here is basically wild-type B cells would dominate the attraction. Okay, and quantitatively that's also the case. We can see the B cells do not express the ligand; they are less attractive to the T cells inside the GC. That we can verify this is the case. So now, with the receptor expression in the T cells, ligand expression on the B cell, particularly under the stimulation by the T cell help, we can see there is within the apparently very chaotic German center environment and a rather deterministic outcome of positive selection, there could be a following process that underlie this translation. Is that the B cell can express CCL22, and if they receive a pretty good T cell help, they could produce more CCL22, and that make the B cells become sort of highlighted even after they break up with the T cells, and that can lead to the attraction of multiple T cells toward these B cells, and that will focus the T cell help to these B cells to be positive selected. So if the B cell acquire pretty good the receptor, then you will let this positive feedback to run and to allow this B cell to quickly select it. So, but of course we couldn't fully visualize the whole feedback cycle just by imaging yet.
But there are quite a few interesting predictions that can make with the chemokine-driven intercellular feedback. If this is really operating, then we should know, we should see the German center B cells that have higher affinity express more CCL22. And to verify that, we use a transgenic BCR that is called BU180 high that has a pretty high affinity for the hapten NP. Then this is the binding intensity on these B cells from the germinal center. If you have polyclonal B6 mice that are following NP protein immunization, there are cells that are binding to the similar intensity than like a BU1 high, then there are cells that are really binding low. So we separate these two populations as a high and a low affinity, and we can verify they're at the BCR sequence level, the particular signature mutation called W33L, that mutation will increase the affinity, and indeed it's very enriched in the high affinity ones, but not so much in the low affinity ones. So this is really a high and a low affinity. Then we compare these two, and then the CCL22 level is indeed higher in the high affinity ones. Then the reciprocal should be true. The CCL22 high cells in the polyclonal germinal center should be of higher affinity due to selection. And to test this, we made a knock-in mouse reporter with a TD tomato behind the CCL22 locus. Then in these mice, after immunization, we can clearly see there are cells that express TD tomato. Then we can sort these cells together with the cells that are most likely do not express much of a CCL22 as the high and the low part, then to measure their affinity by sequence their BCR. So what we can see is the CCL2 high cells contain about 90% of W33L compared to a 63% in the low, suggesting is indeed there's a correlation. And it's important to point out that the CCL22 high has a lower overall mutation load compared to the CCL22 low. That means this result is really a selection rather than the B cell just staying in the germinal center for long enough to mutate more. So with that correlation, then one should consider in the germinal center, the B cell need to compete with each other. The ones that receive help should quickly upregulate their CCL22. Or in other words, there is in the time domain, there should be a pretty tight correlation in terms of the affinity and the help amount and the CCL22 amount. So the prediction is if we acutely reduce the help inside the germinal center, then all the B cells should downregulate the CCL22. And to test this, we immunize the CCL22 knock-in reporter mice. Then we acutely block the CD40 ligand. As I mentioned at the beginning of the talk, that's one of the most important help factor, and also what we saw that can stimulate the CCL22 at the beginning. Then to look at the TD tomato positive cells and the negative fraction in terms of their CCL22 expression. When we sort these cells out, with this acute blockade, we can see there is about 200-fold reduction in terms of CCL22 expression. The reason that they are still TD tomato positive is because the half-life of the fluorescent protein is much longer than the transcript level of CCL22. And the converse is also true. If we deliver exuberant T cell help by infusing the peptide antigen that the T cell recognizes, in this case it's OVA, then the TD tomato positive cells will upregulate their CCL22 by about 200-fold following 4 hours stimulation. That tells us there's a really very, very tight correlation in terms of B cells' ability to produce CCL22 and their affinity, and the help, the recent past of help they have received. Then, if this is really close to intercellular feedback, then if we take away this chemokine, the prediction is then affinity-based selection should be still can go on, like happen in the random mode, but it should be slowed because a random always is less efficient than a facilitated process. And to test this, we created a MUMT bone marrow chimera with the CCL22 17 double knockout, just so all the B cell cannot produce the chemokine, and to immunize these mice to generate the germinal center and look at it over time. So in the wild type GC, then the B cells can produce the chemokine, can construct this feedback with the T cell. Then you see this over a week period of time. This is the evolution of their high affinity mutant. And without the B cell ability to produce the chemokine, and this is the level of affinity maturation. It's clearly slower than the wild type condition. So this chemokine expressed by the B cells is required for the efficient germinal center selection. And in correlation, this is the only slide that relates to the output, is that we know the germinal center underlie the high affinity plasma cell compartment that eventually reside in the bone marrow, and we can also see correlation with this a reduction for affinity maturation. There is a reduction for bone marrow plasma cells in this case. The final prediction is if we force the knockout cells compete with the wild type cells. Now, it's as if these cells would have a birth defect in terms of ability to attract the T cells. So numerically, these cells should have a less efficient contributing to the eventual product of the germinal center reaction, which is the bone marrow plasma cell. But whenever these cells end up in that final product, they should have a higher affinity because they really should work harder in order to reach that destiny. And to test that, we made a 1-to-1 mixed bone marrow chimera by either knockout with the 45.1, which is wild type in terms of the chemokine, or the wild type CD45.2 with the CD45.1. In this case, indeed, if you look at day 21 bone marrow plasma compartment, there is a reduction if the B cells cannot produce the chemokine, and more importantly, for the knockout, when they reach the bone marrow plasma cell compartment, they contain much more frequent high affinity mutant, indicating indeed they have to work harder, and there's a affinity compensated output from the disadvantage of the germinal center cells. I think this is the last data slide. We have begun using this mouse data to look at the human. In the human tonsil, we can verify the helper T cells do express CCR4, and the germinal center cells do respond to CD40 stimulation by upregulating CCL22. And by single cell RNA-seq, we can see the fraction of a germinal center B cell in the human that express CCL22 exhibit a signature of CD40 signaling. So very likely the same positive feedback also operated in the human system. So to summarize, we think from a parent, I think the reason why it's so dynamic, we can spend another talk to discuss that, but the bottom line is no T cell can stick to a B cell for very long, and force the system have to sequentially interact, but yet it have a deterministic outcome. We think that this determinism is really driven by intercellular positive feedback that are operated by CD40 ligand and CCL22. But that comes to, I think, one of early work that also He Ping pioneered when he was still in the lab is there's a contact-dependent positive feedback as well, is that the T cells recognize peptide MHC from the germinal center B cell will deliver CD40 ligand. Now CD40 signal in the B cells drive survival and proliferation, but also at the same time upregulate ICOS ligand that optimize calcium signal to allow better regurgitation of CD40 ligand from intercellular store for the B cell to use. So combined together, whenever the B cell have a higher affinity of BCR, it utilizes two positive feedback to have more likely to be positive selected. One is based on cell contact based, but once they dissociate, the B cell can still attract other T cells from a distance using the CD40 ligand and chemokine positive feedback. I will skip this slide for the sake of time. I think I have mentioned Liu Bo is really the person who did the most the CCL22 work, and I'm very glad to have a vibrant group of graduate students, postdocs, as a colleague in the lab. Hopefully, this is the founding story. Thank you very much.
Okay, so thanks, Hai, for the beautiful talk. We have time maybe for 2 questions. It's a really beautiful movie, and you see the T cell and B cell come together. So what's known about the physical synapse of these 2 cells that are contact? What's mediating their interaction?
Right. I think by the time they have I think in the germ center, it's very, very rarely, if ever, there is a RV2 Kupfers type of synapse that are very stable. But rare is not like impossible. So it has been observed by Jason's sister. There are some, I think it's lasting for maybe, I think it takes 5 or 6 minutes before they can really organize the classic immunological synapse. But that doesn't mean below that time there's no functional interaction. In fact, I think Mike Dustin has been proposing the synapse concept. I think it's what we see. It's actually from early work from He Ping and his colleague. We call it entanglement. It's like the surface area between the 2 types of cells will scale to the ligand density on the B cells. So I think that makes sense in this context. Is the B cells require some contact signal, including CD40 ligand, and that naturally happens in the membrane. So the fraction of the membrane that can be invested into the B cell is what B cell need, and also the vesicular transporter within that interface, rather than to organize PKC theta in a or cytoskeleton in such a perhaps a more like a killer use, because you want to seal that contact to kill a target cells. And it take maybe longer. So in this context, I think Synapse type of a more dynamic organization make more sense.
Okay. Hi, really fascinating story. A question is, so both B cell and T cell are on the move actually. So do you think like CCL22 help B cells attract T cells, right? So do you think that the reciprocal attraction going on?
That's a great question. We don't think there is a risk, because well, I cannot rule that out. We don't have any evidence T cell attract the B cells. That would make things if they. I think we are used to one side chase the other. I think in real life when you date, it's also true. Otherwise, it would be very complex. But the B cells, when they get in contact, there is a reciprocal. The B cell will reorganize so that's I didn't have time to talk about this. So the B cell will reorganize their cytoskeleton to let it become very passive, to let the T cell to test their ligand density. So there is a reciprocal but T cell take the lead. It's when the T cell touch also emanating from the CD40 receptor will loose up the B cell cytoskeleton, and the B cell becomes smoother. So the T cell could really differentiating the peptide C density on the B cell side. So in this way, there is a reciprocal process that.
So yeah. So another question that in the problem with chasing, like you for T cell, like you have multiple potential targets, and they are on the move, and they are sending a signal, but that signal is diffusible. So that can create a very complex situation. So, so like the simple chemotherapy, you have a trail of a gradient.
So the theoretical prediction from your question is, that's a great question. Is it the work distance for this chemokine need to be very short, and that is enforced by all the chemokine degrading and antagonistic receptors. So there are receptors that bind chemokine but cannot signal and internalize, remove the chemokine from the environment. So we do think if the B cell is just pumping out the chemokine, that would basically equalize the field and there's no attraction. And so we are testing the certain chemokine receptor in that function that was our prediction is that will screw up the selection. Yes.
Okay, so with that, let's thank Dr. Hai. Okay. Okay, so our next speaker is Professor Yang Xingfu, who very recently joined Tsinghua University.
So before moving back to China, he was a distinguished professor in UT Southwestern in Dallas. So Dr. Fu is really an international renowned cancer immunologist who has pioneered lower therapeutic approaches against cancer. His laboratory has also made really important contributions to the understanding of gut mucosal immunity, including the function of lymphotoxin pathway as well as the role of gut microbiota and IOCs in IBD and other diseases. So today we'll talk about the next generation of protein vaccine against viral infection. We very much look forward to hear this talk here from Professor Fu. Let's warmly welcome.
About 10 years ago, 7 immunologists from US come to China in part to help science, Chinese Academy of Science build up infection disease immunity and center, and also try to figure out a way to help build up some program for antivirus infection. As a limited time, we only able to and limited resources, we try to figure out whether we should focus on HBV. In taking several years to build up the animal model and also try to figure out what is the problem with many clinical trials for the HBV. As you probably know, most of the therapeutic vaccine trial in China and also other countries basically fail. Despite a lot of effort put in there. And the problem is a lot of potent vaccine are not very potent to induce a therapeutic antibody to control the virus, especially to break tolerance. Another problem is once you use a protein mixed with adjuvant commonly used is alum, the immune response is not very strong, and basically you lost the CTL. And for HBV, you do need to have generate a good antibody and also CTL. So we are looking around new adjuvant that can do it, but most new adjuvant are very toxic. And that's why in most protein vaccine trial, even in the COVID-19 era, still focus mostly use alum instead of using GSK adjuvant and other one, which is very highly toxic to, as a preventive vaccine, become a problematic. So the first part of the story I want to tell you about how we developed the initially HBV vaccine and dealing with the hosts that are already either T and B cell exhausted or tolerance to the antigen. So for the HBV virus, actually most virus as shown as a pseudovirus, and only have less than 1% that they have this so-called intact DAN particle. And we looking at picture and wondering whether that is including in human mouse model where the PS1 is always basically is a receptor binding domain and express much lower level than HBV. And we know the antigen level in the HBV is surface antigen is quite high and induce tolerance. So we built up this model using AAV HBV intact entire viral genome, and in fact, and we noticed something interesting is once the antigen level is higher, it always induces strong tolerance that when you give a traditional HBV vaccine, this is a surface antigen vaccine, and we do not able to break tolerance and the virus antigen and or induce any antibody response. However, if the antigen level is lower, then you give a conventional vaccine that is a preventive vaccine, basically surface antigen plus alum that most young people now use as a preventive. And it actually can clear the virus. Not only they can
reduce virus antigen, but most important is can induce HBV, anti-HBV surface antigen antibody. And that basically considered as a clinical cure, where you can generate the endogenous antibody response to neutralize surface antigen. So we wondering whether we could actually looking into this issue further, maybe go the other way around to ask a question, whether PS1 as a receptor binding protein is actually better target. We know that everyone using, most people using this surface antigen as a vaccine may not be able to induce a immune response. And indeed, if you use surface antigen as a vaccine and use ALM, and you see this in wild-type mice, it generates a good response, antibody response, but in carrier after 21 days, you are not able to generate any response at all. And same as a T cell response. However, if you take a PS1 as a vaccine, which we think is the weakest link in virus in terms of vaccine, and carrier now responds very well. And that's also in the T cell response. And if you take a PS1 with strong adjuvant, and even carrier, you can reduce the PS1, and actually even surface antigen also partially reduced, and you are not able to detect virus DNA in the serum, because of all the viruses have a DNA in the serum that actually was packed by PS1 as a whole virus. And even you can take a serum from here versus serum from other place. Only serum from here can actually neutralize the virus. This is the Li Wenhui's cell system, where you can have live virus infection, and you can see a clear neutralization by using PS1 as a vaccine. You can induce antibody that can neutralize live virus infection. And that's very important for us, because our goal is to generate the endogenous immune response to neutralize virus reinfection, which is very important in human case. So in that regard, we also realize we cannot using CFA, IFA, all these strong adjuvant to the human, because even mouse will complain too. So in human, that's too toxic. So how do you develop a vaccine that can be used widely in the children without clear toxicity? And we figured out all these different vaccines. There is a common cytokine that are important for antigen presentation is interferon. So interferon could basically increase antigen processing and presentation and DC maturation, and also increase B7 expression and Cas1 expression. And also interferon can driven the Th1, TC1 media response. Otherwise, when you mix a protein with adjuvant, like ALM, the CTL response disappear or greatly reduce. So-called in the older story, we call the cellular immune response was suppressed by the humoral response. Now, another important feature we figured out that a couple of years ago is a receptor binding domain. It's better to form a dimer, and they are much better than monomer, as I will show you soon. And we think the dimer formation is more natural to attract high affinity B cell and response, and also avoid this so-called antibody enhanced problem. And as the size of vaccine as a dimer also increases size to allow the antigen go to drain lymph node and allow more antigen go to CT and B cell. And FC is also important, of course, from this dimer and allow a long, not only long half-life, but also help the antigen capture through the FC receptor as well. And you can see here that PS1 itself as a monomer, and they cannot actually generate much antibody response. And if you use a dimer with FC, their response greatly increase, maybe 104. But now if you add interferon, it reach to a very high level, and that's very important to break tolerance and neutralize virus. And you can also take a serum and able to neutralize crossbore of various HBV strain that cross to north part of China, south part of China, and also west part of China. There's 3 different types. And now more important is you can see this is a carrier mice that fail to respond to any type of vaccine if in the alum. Basically a protein vaccine with the alum. But here you can see we have this vaccine. The only part is this the one that have interferon. You can see this is a dimer with interferon versus the dimer without interferon versus just a monomeric one. And you can see that the PS1 is only in this group drop. Okay, and anti-PS1, only this group have persistent for long time. And interesting part, we also see surface antigen starting to drop and DNA also drop. Now, we therefore decided, because even though we figured out this response is pretty clearly to neutralize the live virus, but for many patient and physician, the traditional view is you need to reduce surface antigen and induce anti-surface antigen, and therefore they think it's a clinical cure. So we decided to add additional vaccine. This is traditional vaccine that you can buy from any pharmacy store, and you using these 2 combination. And as a vaccine, we found out that not only you can generate anti-PS1 antibody, and also reduce the virus PS1 antigen. But more importantly, now this is the first time in our system that we are able to generate anti-HBsAg antibody, which is a clinical indication. Right now, most of the clinical trial not able to generate anti-surface antigen antibody. And that's why when you stop treatment, the virus often relapse. And this is also only one with a combination you are allowed to reduce this dramatically over the time. So in that background, we come into the last year, around May, we realized current vaccine developed globally have major problem, either not induce strong titer, neutralized titer, or too toxic, including mRNA vaccine. We think the side effect is quite significant. So we decided to try some effort, convert this virus basically using a receptor binding domain from the COVID-19 and convert this to this format. And basically we just replace the RBD from HBV to COVID-19, and this is a structure we estimate. And basically, we call this super RBD now, and you can see here, if you just have RBD, the response is quite low. But if you use this super RBD, then you get a very strong specific RBD response. And also, they have a strong neutralization antibody, suggests that you have high affinity antibody in the system. And basically, that antibody is about 3 times higher than patient's serum level. And you can also see how low the vaccine you can use. You can use 0.01, you still generate a very strong immune response. This 0.01 microgram, and you can easily generate 1 gram per liter of antibody through CHO cell. And so you can see that if I produce 2,000 liter you can supply entire Zhejiang province. And you can also see this, the RBD as a monomer is not a very strong immunogen. And if you form the dimer, it's better. But if you add the interferon into it, that become much better. This is dimer without FC. This is dimer have through FC form the dimer is more stable, and you can see this trend of increased potency of this. And we know to generate a high-level neutralized antibody is key for this pandemic. And even we use very low level, this 0.01, you can generate a very strong response when you have interferon in the system. And basically, we found out that this is the level of increase to all the way to interferon that add the additional T cell epitope. Now, as I mentioned, this is extremely low level. You can generate a very good response if you use super RBD, but regular RBD, basically you see much limited response if there's anything there. And also, even you use single shot, you probably know from even mRNA clinical trial, first, a single shot do not generate a protective immune response because they don't have enough antibody through single shot. But we have this single shot, we can generate actually very good antibody response, including neutralized antibody response. And second, very interesting and important feature is you don't need adjuvant here. This is a vaccine without additional adjuvant into the system. And that's also important because currently in China, there is no endogenous domestic industry can produce very good adjuvant. Even including alum, is mostly from import. So you can imagine if someone decide not to give this adjuvant to Chinese company, it will be a disaster because protein cannot generate the immune response without adjuvant. And this is to show you using a pseudovirus neutralization. In some of the experiments, we actually work together with Zhang Zheng here that help us to compare with the patient's sample that his lab generated. And for mechanism, one important thing is a super RBD can reach to the lymph node a much higher level than RBD itself. Because of the size, we think is important. Size always matter. And also, we found out that dendritic cell can uptake antigen much more efficient if you have additional this engineering system, especially with interferon. It could help dendritic cell uptake antigen and help them express CD80, which is a key co-stimulatory molecule for this, and also help T cell in drain lymph node. They are also increase germinal center. So there's an important feature of this second generation of protein. And also, if you look at if you put with just RBD, you generate some Th2 response, but you cannot generate the Th1 response. And we have both Th1 and Th2 response. And also very important, we generate very strong CTL, which most protein antigen that when they are in the alum, they cannot generate the CTL. We also show this Th1, Th2 T cell from intracellular cytokine staining as another evidence to show that with additional interferon, you increase both Th1 and Th2 response, and also CTL. Not just increase neutralized antibody. Now, if you look at this, this is a monkey study that we have done together with the institution in Chinese Academy in Yunnan, and we found out that you can generate a very strong, with both low and high antigen, One is 10, one is 50. You can generate a very good response in both regard, and they are very high titer of neutralization. Average is around 5,000. And the monkey, after high dose challenge, and they don't have a high temperature. And most important is if you take this picture, for example, from tracheal or nose swab, and after first day, you are not able to detect any virus. And that's very important because the virus can detect here means they can actually spread to other hosts. And our vaccine, because produce very high level of antibody that could rapidly neutralize the virus, therefore you are not able to detect the virus in both in a high and low dose after first days. And in the lung, obviously, you don't see much at all. And this one without adjuvant, you can see in this monkey study, they could last more than 200 days and still able to detect quite a significant level. And so humans supposed to be even longer. And now there's another very interesting feature of this vaccine because you don't need an alum. So you can actually have taking the 10 microgram of vaccine nose drop, basically 点鼻子一下就行了. And you can see very high level of IgA. I was actually the first people to test this myself. I asked Li Zhu pharmaceutical company after they produced initial batch. I asked them to send it to US. I was the first one in early November. Taking my wife and a couple of friends. We just because at that time we are not able to get vaccine. Even US, as you know, they started later December, early January. Right? So we don't have any source of vaccine. So we decided to, in the surrounding, many people get infection in such a dramatic way, especially in Texas. Nobody, you go to the Costco's, they do not allow you to wear the mask in the early days. They look at you, probably want to ask you, get out of my store if you wear mask. So we have to use a biological mask and to able to go to store in and out without any trouble. So I was the first testing body, and it's very convenient. So once I did, many friends asked me to give this solution, basically put in the water and just put in the nose 100 microliter, and you're feeling very good. So, because I also measure IgA and IgG, and that's why I asked a question about mRNA. Actually, not able to detect much IgA at all. And our system, we can produce, and we found out that always wife produce 10 times more IgA than husband. So we have several pair to do the test, and it's very interesting. Women actually response have more antibody protection than a man. Maybe in evolution, woman is more important. So this is also we studied this in a clinical trial. You can see here in the phase 1 trial, the placebo group is just use alum, and with this one with that. Actually, a side effect is very similar. Not much higher if you use 10 microgram. Is not much higher because you only have 2 microgram interferon, which will not cause any feeling, the response side effect at all. And this is actually real patient phase 1 trial done in Guangdong CDC group, and you can see here that many patients, even with very low dose, and they produce a consistent. If it's high dose, it's 100% even in senior people who are around 70 years old, and they still produce a very high antibody response, and usually it's around 3 to 5-fold compared with the convalescent serum. So titer is high because compared with inactivate virus, they produce usually around 20 for some people. Of course, for many, it's below 20, and they define as 8.
And also, we can use very low dose. And the protection is fast, and for most people, it's about 100%. If even senior is to go to 96% response. And also important feature is it's good for Delta. It's good for everybody. So for Delta variant, we also have a very strong protection. And this is a monkey that after 300 days, we decided to challenge. We found out that you can increase 78-fold to wild type and 53 for Delta, and even for Beta, which is the most difficult one, and we still get very good protection, neutralize antibody as a protection. And they increase level very extremely high for Delta. And that, and this is just tell you how many fold increase over the second dose. And and we found out that for various if you just use conventional V01 is a vaccine. We name that V as a vaccine, also as a victory, and able to produce very high level of antibody neutralize all 3 different type of variant. And even in the real patient, there's several 30-something people from Lizhu want to go to Southeast Asia to do clinical trial, and they cannot detect any antibody even after 2 vaccines. So they use this as a boost, and they get very good response. So I think I should end up here because the interest of time, and I will basically just try to argue this is safety is good, neutralized antibody is good, and also good for senior people, and very effective for various variants, and it's very easy to produce. Okay.
Okay. Thanks, Professor Ku, for that very impressive talk. So we have time maybe for 2 or 3 questions.
Very nice. I just wonder if you can use this for the flu virus as well, like for.
Oh yeah, you can
HA.
make these 2 flu virus and other virus just replace the
RBD.
receptor binding. Yeah, IBD.
I have a question. So for the antibody level, is it the higher the better, or is there a level that it's enough?
Well, if you think it's too high, you just reduce the antigen and save the antigen dose. So I think you need a conventional thought right now. They think it's above 100 neutralization dose. 1 to 100 above is better. So usually you have 100% protection. But once you go down, the protection gradually reduces. So ideally, you want to reach, after vaccine, reach to 100, which is very difficult for right now, most of the vaccine we use in China.
Okay. Another question is that if the immune system spend, more efforts to neutralize the SARS-CoV-2, does it affect the immune system to react to other disease?
Very unlikely you can see significant effect because the antigen response, well, antibody response is very specific. But if you measure the serum antibody, the antibody you talk about is extremely low level compared with other antibody around. So I don't think it will see major impact shift to only to this. You can still vaccine others too.
Thank you.
Yeah. You must have mentioned this. I missed it. So how is the neutralizing antibody levels compared to Moderna or Pfizer vaccine?
I think it's close. We are close to 200. They are close to 250.
Okay.
Or maybe 300. Yes.
Yeah.
So I have a question. So you mean the IPF plus surface antigen vaccine can promote the production of the neutralized antibody in IPV mouse, and you mean the pre-S1 can promote the neutralized antibody?
Yeah, anti-PRIS1 usually neutralize all the
virus particle, DEM particle, because all the DEM particle have PS1, PS2 to form the intact virus. You are the expert on this part, but to make a physician and patient more accept this, we mix with regular vaccine, and that could induce also anti-surface antigen antibody. As you see many patients, if they can see their blood have this antibody, right? Everybody is so happy, but it's extremely difficult to reach to level for previous treatment. Even with all this drug treatment, so far it's very, very difficult to generate anti-surface antigen antibody.
Hey, Yang-Xi. In terms of the IgA, is that a serum IgA or is that a mucosa IgA in the nasal?
Serum IgA. We can reach it if you do a nose drop. The serum IgA increase dramatically, especially for those people who already have previous vaccine.
Sure. But I thought it would be. Did you try like?
Mucosa we also try, but it's very difficult to decide. You know, I have to put in a lot of
that would be
effort to do it.
For public health, that seems to be.
Yes,
because if the mucosa IgA in the nasal.
Yes, so it
prevent
the
transmission.
Yes, that's very important. And the difficult part is globally, in the last 20 years, a lot of effort try to making the drug to using, nasal drop. The problem is how do you define? Because you always take blood to measure it. So it's very difficult to compare with intramuscular injection. Obviously, serum level is higher, but that's a little bit misleading. But there's no easy way for FDA or CDE to decide how to choose.
Okay, so due to time, we thanks Professor Fu for his wonderful talk again. So could we bring Dr. online?
Hello, can you hear me?
Yes. Okay, good. So sorry again for the technical issue. So we very much appreciate for your patience. So could you please continue your talk?
All right. So sorry, I was using 5G at home. Apparently not very stable. And now I will continue the talk, and I use the time to delete a few slides. So I will directly get into the immunology.
As I mentioned that I was a virologist.
Could you please share your screen? So we cannot see the slide.
Okay.
Okay, great.
Can you see that?
Yes.
Okay. So I was a virologist and tried to study immunology as I'm moving from the US to China and have a clean start and have a different perspective on the questions we want to address. The questions about how virus interacting with the host and how host control virus infection. And since I was quite naive with immunology, and we try to look at the immune response more comprehensively and with a high resolution. And one of the challenges, there are many, many challenges, and today Akiko's talk present many questions. And one of the challenge in immunology is to identify the target of numerous antibodies. In each individual, there are millions different type of antibody, and what are those antibodies against? What are those T cells against? Today, I'm going to concentrate on the antibodies. And we try to develop a method that can profile the antibodies in an individual comprehensively. And this is our first effort, and we started last year. And since I just moved to Hong Kong, and we have very limited resource as the pandemic goes on. So here I will show you the first attempt we tried to address this question. And the technology we're using is called messenger RNA display. And it was invented by Richard Roberts and Jack Shustab in the 90s. And here at the bottom of the diagram show the translation process. And as the ribosome move along the messenger RNA, it will make nascent RNA. And the puromycin is a mimic of the end of the tRNA and carries the amino acid, and the puromycin gets into A site and conjugated to the nascent RNA, and then for a covalent bond between the protein and the messenger RNA, and that creates a link between the peptide and with the corresponding messenger RNA. And the messenger RNA can be function as a genetic identity for the protein. And here on the upper part, you can see the similarity between puromycin and the end of tRNA, and that's why puromycin is effective antibiotics and block the chain elongation. Okay. So this creates the simplest form of linkage between a protein and the corresponding corresponding genetic code. So we have used this technology, and we have been improving this technology with messenger RNA display. And here, the experiment we have done about 10 years ago, and we take a protein, we mutate every amino acid of the protein, and measure the affinity. So this protein is part of protein G and bind to IgG. And then we mutated every amino acid of the protein and examine the affinity of the protein to the target. And here is the profile. And when every amino acid was mutated, you can see, depends on which amino acid mutated to, it has very different phenotype. And this generates a very comprehensive profile of the impact of each variant on affinity. And that sets up a foundation. And next, we increase the throughput of this method. And we made a double mutations, pairwise mutation of the same protein, and examine the affinity of the double mutations. And that basically measures the affinity of 500,000 variants to the target pretty precisely. And people actually can use this functional data to de novo reconstruct the protein structure. And two labs did that independently. So the data is pretty precise and reliable. So after the pandemic come, and we said, okay, maybe we can use this to study immunology. ELISA, everybody knows, is one of the traditional methods to measure antibody response and the interaction between antibody and antigen. And we changed the ELISA by incorporating the messenger RNA display. And the procedure is pretty similar to ELISA, except that the antigen preparation is different. The traditional ELISA, you put an antibody, captured antibody on the plate, and then you put in one antigen and then measure the luciferase activity or fluorescence activity. And here, and I want to draw your attention to the bottom of the middle of the slide. And here we have hundreds, thousands of different peptide or antigens linked to the corresponding messenger RNA. So each peptide has the genetic code covalently linked by puromycin with the method I just described a few minutes ago. So we create a large pool of peptides with the corresponding messenger, and which offers two advantages. One is because the genetic code, it can be PCR amplified, the sensitivity is very high, and second, increase throughput. So we go through the ELISA process and watch, and then see which antigen are bound, and by examining the nucleotide sequence attached to the antigen. And so the PCR amplification and next generation sequence and identify and quantify the antigen that have been bound by antibody. So we're using this method to examine the immune response in COVID patient. The first library construct has about 100,000 peptides and each 48 amino acids long. And we covered SARS and all older SARS and all mutants of SARS-2 have been reported by the date we synthesized oligos. That was synthesized in the middle of last year. And we, including all the common cold coronavirus, all influenza virus has infected the human in the last 70 years, and most commonly seen virus. And we including EBV. The blue ones, I'm going to talk about the data. The others, we have the data, but I will not talk about. We're also putting autoantigens, all reported autoantigens, about 1,100 something autoantigens. And after that, we made a second library, which contained all possible variants of the epitopes that we identified. Okay. So here are the patients with COVID. The lab is in Hong Kong, and Hong Kong managed COVID very well. They have good social distance and they have good health care. So most of the patients are not sick. They were identified early and probably infected with low dose, and not many people have severe problems. So we have this cohort of patients, which we longitudinally collected the samples, serum samples from onset, and we identify equal number of sex age matched pre-pandemic serum samples as control. And I will also mention that we have infection model and the lupus as control, and why we use this as control, it will come up in a few minutes. So here are the baseline data, and to demonstrate the method, EBV are infecting majority of the human populations, and we have EBV epitopes, about 1,500 peptides about EBV and its variants. And here are the IgA and IgG response against EBV epitopes. And this is representative of one patient, and it turned out to be very stable. Among patients are stable at different time points of each individual patient, which is a huge contrast to COVID and antibody response against COVID. So here is the data on the COVID. And so each dot, a square, the color indicates the intensity of the antibody response against the peptide. And each line, each line here represents one peptide, and then the color indicates the strong, how strong the patient's response. So here are the 54 patients, each how they respond to the peptide is indicated. And here are the pre-pandemic controls. You can see pre-pandemic people also has antibodies against certain peptide, but at a lower intensity and a much lower number of people had, which is consistent with during pre-pandemic period, and quite a lot of people infected with common cold coronavirus. Some of them share sequence homology with the SARS-2 virus. So you can see we can identify the difference between the COVID and pre-pandemic, and we will look into the details. As I mentioned, that we included all mutants reported during that time, and the mutant usually is a one amino acid difference, as indicated. Here's amino acid alanine to A to D change, and then the antibody response against the wild type has a very clear peak curve during the middle of the peak of antibody response, but decay very fast. And this patient is not recognizing the mutant. So the mutant potentially can escape this recognition. And the same with an antigen in N. This is antigen on epitope on S. And so we can look at the antibody response against all reported mutants, and there are 26,000 mutants being reported at that time point. And we can examine the potential to escape recognition against all those mutants of concern, or variants of the concern, whether they have potential to accept antibody and that epitope against that epitope. So this is really high resolution, and actually all those data was obtained with 1 microliter of serum. And I have mentioned that we use EBV as reference point, and you can see in one individual, antibody response against EBV did not change after infection, within 4 months after infection, the EBV antibody did not change, which also demonstrated the reliability of the consistency or the robustness of the assay. And in contrary, the antibody response against SARS-2 epitope varies quite dramatically in 4 or 5 months after infection. You can see one individual, the antibody response profile changes. And this is not one particular individual's case, and I think the data is similar from other people, and the antibody response against individual changes, and there are many series, and Akiko mentioned some of the possible series. And so the antibody response is quite dynamic over several months after infection, and here is a summary of the dynamic pattern of the antibody response. And this in the middle indicates the weeks after virus infection. And the color, the yellow color indicates the frequency of the antibody being positive against this peptide. And you can see, for example, this patient against this antigen was recognized very early on, but it decayed relatively soon. And there are certain epitopes has persistent antibody response. It's highlighted by the red line in the middle. And those are the epitopes constantly being recognized by majority of the people. And while the other antibody response against other epitopes are more transient. So the next stage, we synthesize variants of the epitopes that are commonly recognized by majority of the patient and do a more detailed analysis. We made the variations of single amino acid change on the peptide. So if you look at the bottom of the slides, 48 amino acid were changed to all other possible amino acid and made a pool of mutants. So here show the details of the antibody response. So here is an example of one peptide and 48 amino acid. We made all possible changes, and then all the variants, how they recognized by the antibody was measured. So the red indicates that variant is highly recognized by the antibody, and the white means the binding is weak. So this generates the extremely detailed fingerprint of antibody binding capacity or affinity specificity. So we summarize those data by looking at the antibody, the frequency of the peptide being recognized by each individual as a surrogate of how broad the antibody response is. And the yellow color indicates more variants was recognized by the antibody, and the blue ones less recognized, less variants was recognized. You can see the broadness of antibody response changes over time, and some people has very broad immune response at early time points like this one and the decade, and some people has lower broadness and then increase. So you can see the antibody response extremely diverse. And in contrary to the traditional immunologic assay, you only have one indicator, which is the titer of neutralization or titer against one recombinant protein. And here we are presenting a very detailed fingerprint of antibody response against many, many peptides during the infection. So this brings up a lot of more questions about how do we characterize immune response, and here it provides tremendous information. We definitely need to work with the data and the AI person to summarize and analyze those data. So here another detailed analysis of the data. SARS-2 has this peptide, and common cold coronavirus has a homologous peptide. And you can see on this side, on the left side of the screen, you have the pattern of how antibody recognize SARS peptide and all its variants. And also, since this has homology with the common code, and then we try to distinguish the fingerprint and the variants of the common code peptide, you can see they have different patterns. So this will be a challenge for us to how to recognize those such a fingerprint and memorize. Okay, so previous several slides, I introduced how we analyze the antibody response against viral epitopes. And next, I will present the data on autoantigens. And in this pool of epitopes, we included 1,100 autoantigens and tiled array against all those antigens. And here is a summary of those antibody response. And horizontally, each line represents one patient, and then vertically, each dot represents one autoantigen. And on this side, antibody response against in COVID patient, and here the antibody response in pre-pandemic control. You can see pre-pandemic control, each person has a little bit of autoantibody response, and there are no fixed pattern. And then in the COVID patient serum, there's increased antibody response against more autoantigens after COVID infection. And the classification of those autoantigen recognized by those patients are neurological receptors and coagulate proteins, which is consistent with the increased risk of patients getting those long COVID disease. So we're verifying the proteins we identify. So I have mentioned that we included infectious mononucleus patient and try to see whether there's a similarity. And next we're going to look at influenza virus infection, whether other virus infection, respiratory infection induce similar phenotype. We hope not, and it's more specific to COVID. And we will see the result very soon. And so you can see that infectious mononucleus also induce extensive inflammatory response and induce less autoantibodies. And the autoantibody response of COVID patient actually more similar to lupus patients. So this is consistent with the general report, and in COVID patient, autoimmune response was induced. And also, the autoimmune response also has very different dynamics. This is one patient, and response against one autoantigen is this curve, and different patient, different curve, and with different peak and different pattern. And so if we look at the peak of each individual person against each individual protein, and then summarize, and then the color indicates the more patient has the peak at which time point. And you can see that majority of the patient against the majority of autoantigen are peaks as between 2 and 3, 4 weeks, 5 weeks. Okay, and so this is making sense. But also there are certain antigen have much delayed peak of the autoantibody response. Okay. So I will summarize the new technology we developed, and the platform technology enable comprehensive precise detection of target specificity of antibodies, and we can reach close to 1 million targets with 1 drop of blood, and this will be utilized. It can be utilized with other clinical settings in addition to infection. We are currently looking at the vaccination, at how broad the antibody response and how strong the antibody response during vaccination. And in the next just 2 minutes, I would say how we're going to use the immunology, the new platform, to integrate with the virology we have been doing. We have mutated every amino acid of viral genome, so we have done that with HIV, HCV, and influenza virus, and I did some with the Zika virus as well. And here is the most comprehensive work, which will mutate every amino acid of the influenza virus genome. Every amino acid, which we have the data over 97% of amino acid, and a few percent the data not reliable. Okay, so based on such detailed genetic functional map of the virus genome, we select 8 mutants, and each mutant decrease the virus replication in the presence of interferon and by induced more interferons. Each mutant induce interferon a few-fold, and we select 8 mutants combined together, induce interferon production about 50-fold. This is extremely similar to the parallel with what Fu Yangxin has just mentioned, that by increased carrier interferon into the site of the antigen, and the antigen will be better recognized and induce much strong immune response. So we engineered this virus, has 8 amino acid change and induce 50 times more interferon. That become a spectacular vaccine, induce robust T cell and antibody response. And here is the T cell response. Actually, the virus replication was reduced about 3 logs, and pretty much the replication never took off. And the virus induced the same amount of T cell response, both CD4 and CD8 response, and established memory T cells as efficient as a full-blown and full-scale virus replication of wild-type virus. So we engineered a virus that's much more immunogenic than the wild-type virus, and by induced more interferon, the virus replication was inhibited, but it induced very strong immune response. So the virus we construct maintain virus fitness, replication fitness in the absence of interferon, both in vitro and in vivo, but it induces robust antibody and T cell response, and it can block the infection of homologous and heterologous virus infection in animal models, in mice and in hamster. Just got blocked. Okay, another animal. So it's broadly applicable to vaccine development and against the pathogens, and we also can use this principle to induce immune response against tumor antigens. And this work was done by Yusen Du. Now Yusen Du is having her own lab at Zhejiang University, and she is also interacting extensively with people in Westlake. And I'm sure she's going to make a great progress on using the virus to induce antigens in tumors. Okay. So I have presented you the approach that we have developed to characterize antibody response comprehensively and with great details and precision, and it generates very dynamic view of the immune response in a systems way. At the same time, we have generated a pool of virus, and we can examine and tracking the fate of each individual virus in the population. So traditionally, virology and immunology are being studied individual antigens, individual T cells and B cells, subtype of B cells, T cells. Now we can look at both populations, and which will enable us to look at viral host induction in a much more comprehensive way. And I'm very excited to be able to combine those two systems approach and look at the interface between virus and immune systems. So I'd like to acknowledge the people did the work, and Yusen Du was the one led the influenza virus work, and also initiate the work on messenger RNA display in collaboration with Anders Olsen and Nicholas Wu. And it was in collaboration with Richard Roberts, who initially invented the messenger RNA display. And the antibody response was led by another postdoc, Xiang Zimeng, and in collaboration with outstanding clinicians and microbiologists at Hong Kong U. So I'm okay. I'm ready to take questions.
Okay, so thanks for this very cool talk. So we only have time probably for one question, if you are not hungry.
Do you have some idea why the COVID-19 induced antibody dropped so quickly compared with other viruses?
So I think, it's just speculation. I think the virus has a way to deregulate the immune response. And without creating proper microenvironment for the antigen to be properly recognized, or the T cell, B cell was not properly trained or educated at the site of infection. And presumably, one or two cytokine was too high or too low, of many cytokine was too high, too low, and some T cell was not properly trained. So cannot establish the long-term memory. That's my speculation. And willing to learn what's going on. Yeah.
Okay, that's great. So I think that's thanks again for all the speakers this morning for their very wonderful talks. And it's time for lunch.
讯设备关闭或置于静音状态。谢谢您的合作。
Ladies and gentlemen, the event will begin shortly. Can we please ask for you to kindly take your seats? We would like to remind you again to switch off all mobile phones and put electronic devices to silent mode. Thank you for your cooperation.
Okay, so welcome back. So I think it's time to get started. So I'm Zixun Liu. I'm from School of Life Sciences, Westlake University. It's my great pleasure to introduce the next three speakers. The first one is Dr. Feng Shao. I mean, most of you may familiar with Dr. Feng because he's really like a good scientist in China. So I mean, his work, and he got his bachelor degree from Peking University, I guess, and then he moved to US and get the PhD from University of Michigan, and he did his PhD work in Jack Dixon's lab. I mean, Jack Dixon is a really famous biochemist. So he started his own lab at NIBS. And the first, he was focused on the bacterial effectors, which target hijack the immune system. And I think there was next he studied a lot of like innate immunity, especially particularly in the like paraptotic cell death, which is caused by the bacterial infection. And finally, he figured out which the family of called gastamine protein, which can mediate the apoptotic cell death, which is really important to antibacterial. And now, recently, he discovered a lot of like work about the gastamine protein functional of the anti-tumor immunity. So today's topic will be apoptosis in antibacterial and anti-tumor immunity. Let's welcome Dr. Feng.
Hello, everyone. Can you hear me clearly? Okay, I would like to thank the colleagues at Westlake University to invite me to give a talk here. Unfortunately, I cannot be there. So today, what I will do is I'll introduce pyroptosis to you in the first part, and then I will talk about our recent work on the function of pyroptosis in antibacterial immunity, and then I will switch to the role of pyroptosis in anti-tumor immunity. So many of you have heard a classical cell death called apoptosis. So in apoptotic cell death, you see the cells shrink and fragmented into small vesicles. We call it apoptotic body. The key feature for apoptotic cell death is the membrane integrity is maintained, and there's no inflammation. So apoptosis is more like a immunological silence form of cell death. On the right side, you will see the cells undergo pyroptosis, and you see the cells did not shrink. Instead, it was swell, and you see balloons coming out from a plasma membrane. Eventually, the pyroptotic cells undergo membrane lysis and then release cytosolic contents. So pyroptosis will trigger very strong inflammatory response in the body. So historically, pyroptosis was originally observed in bacterial toxin or bacteria infected macrophages like anthrax lethal toxin or Shigella infection that was observed in the more than 30 years ago. So that's very key, that pyroptosis was originally observed in bacteria infected macrophages. So pyroptosis indeed is a host immune response to bacterial infection or toxin intoxication. So in innate immunity, we know that you need a sensor to detect the infection. So that is called in the pyroptosis pathway. That is called the inflammasome pathway. So we have this so-called canonical inflammasomes. The canonical inflammasome is a multi-subunit cytoplasmic complex. Within the complex, there are null-like receptors were directly or indirectly recognize microbial products. For example, the NAP-NRC4 inflammasome, which can directly recognize bacterial flagellin or components of the type 3 secretion apparatus. We also show that the pyrine inflammasome can indirectly sense the activity of bacteria, many bacterial toxins. So the canonical inflammasome will recruit the procaspase 1 and then instruct the procaspase 1 to undergo autoproteolytic processing to form this active form. Active caspase 1 will process interleukins, interleukin 1 beta and 18, and also trigger stimulate the cells to undergo pyroptosis. we have another pathway called a non-canonical inflammasome pathway. And this pathway, caspase 11 in the mouse, caspase 4 and 5, which is the counterparts in human, will directly recognize lipopolysaccharide within the cytosol space. So we not only have TR4 on the membrane for recognizing LPS, we also have caspase 11, caspase 4 and 5 within the cytosol to directly recognize LPS. And then that will induce oligomerization of caspase 11. Active caspase 11 will only trigger pyroptosis without cleavage or processing of interleukin-1 beta. So common to these two caspases is the cell death, pyroptosis. So what is the underlying mechanism? Quite a few years ago, we through genome-wide CRISPR-Cas9 screen, Jianjin Shi and Yue Zhao in the lab, they identified gastamine D that is critical for both caspase 1 and caspase 11 mediated pyroptosis. So gastamine D functions like a switch. You have gastamine D in the cell, you have pyroptosis. You don't have gastamine D, there's no pyroptosis, at least for caspase 1 and caspase 11 pathway. So the gene encodes a protein of about 500 amino acids, 2 domains, N-terminal domain, C-terminal domain. And these 2 domains bind to each other that keeps the protein in the inactive state. And within the linker region, there's a caspase cleavage site, FLTD, and the cleavage will release the N-terminal domain. The N-terminal domain will oligomerize when encounter membranes. So, and then form pores, oligomerize the N-terminal domain forms pores. You see the multiple pores formed on this liposome structure. And the pore is quite large. The inner diameter is about 22 nanometer and is sufficient for I1 beta to get out of the cells. So in this field, it's now a common, it's very well accepted that this large pore is actually the conduit for mature I1 beta to get out of the inflammatory cells. So, and this region indeed is a membrane inserting region. So when you have multiple pores formed on the membrane, the cells will undergo swelling and the disruption of osmotic potential, eventually undergo membrane lysis. So last year, we also further figured out the biochemical mechanism of how do caspase 1 and 11 recognize gastamine D. So traditionally, it was believed that all the caspases recognize the cleavage site residues, like FLT, in the case of gastamine. And through biochemical and structure studies, we found that caspase 11, as well as caspase 4 and caspase 1, the active caspases are dimer. They use the exosite to recognize the C-terminal domain of gastamine D. So there is a hydrophobic interaction here. And this interaction actually renders the tetrapeptide independent cleavage. So if you make a mutation in the FLT sequence, you still get very robust cleavage of gastamine D by caspase 1 and 11. So this caspase and substrate recognition mechanism is very unique. It's different from all other caspases. So if you are interested in this story, you can read this paper published last year. So pyroptosis indeed is downstream of the inflammasome pathway that is part of our innate immune protection mechanism. So if we knock out the gastamine D, we'll knock out or block or diminish pyroptotic response in response to bacterial infection. So there are two consequences. Under normal infection situation, if you knock out gastamine D, the mice are more susceptible to bacterial infection. That illustrates the protective function of pyroptosis. On the other hand, when the infection goes systemically, when you have bacteremia, if you have bacteria in the circulation system, and then that will trigger so-called septic shock. If we knock out gastamine D or caspase 11 in the mice are become resistant to really high dose of LPS-induced septic shock. So I'm now showing you some examples of how do gastamine, the pyroptosis pathway mediates antibacterial immune function. If we infect mice with this gram-negative bacteria called Chromobacterium violacin, and this bacteria is recognized, recognized by the NAP-NRC4 inflammasome. So wild-type mice can can contain the infection, so that the wild-type mice can survive out of the infection. If we knock out caspase-1, NRC4, or Gastamine D in the mice become susceptible. And you see the mice died within a few days upon infection. And you see, if you measure bacterial load in the liver of the spleen, you see that the knockout mice have much higher bacterial load in chromobacterial load in their liver and the spleen, because the knockout mice lose the knockout mice can lose the inner immune protection mechanism. They cannot limit the control of the infection anymore. Very similarly, with the Francisella infection, this bacteria is recognized by the M2 inflammasome. You see very similar response. Wild-type mice can contain the infection, can survive out of the infection, but the caspase or Gastamine D knockout mice are much more susceptible. They succumb to the infection, and you see the bacterial load is the same story. But in these two cases, you might recall that caspase-1 can also process interleukins, and the mature interleukin require Gastamine to get out of the inflammatory cells. So if we knock out Gastamine D, we cannot attribute the function or the phenotype solely to the defective pyroptotic response, because knockout Gastamine D also block IL-1 beta or IL-18 secretion. So we don't know whether it is a pyroptosis itself or pyroptosis together with cytokine production that are important for the mice to limit the infection. So then we turn to another bacteria, another type of bacteria, such as Shigella or Burkholderia. Those bacteria, they are intracellular free living. They don't live in the membrane bound vacuole structure. They inevitably expose LPS to caspase 4 and 11. So you would expect that bacteria like Shigella or Burkholderia infection will stimulate a very robust LPS-induced pyroptosis pathway. So this was a story published a few days ago, and this work was done a postdoc fellow Zilin Li in the lab, and it's actually a very interesting story. So if we infect mice, here we are doing the mouse infection. Infect mice with Shigella or Burkholderia, both are intracellular free living, right? In the case of Burkholderia infection, if we knock out caspase 11, the mice are susceptible to Burkholderia infection because the mice cannot lose the LPS sensor, cannot stimulate the pyroptotic response. However, for Shigella infection, wild-type and the caspase 11 knockout mice, we don't see any difference. Both strains, both types of mice succumbed to Shigella infection. We then did a series of work and figured out that the bacteria Shigella encodes an inhibitor for this pathway. The inhibitor indeed is a toxin, is a type 3 secretion factor we call OSPC3. If we remove OSPC3 from the bacteria, now the mice can contain the infection, can survive out of the infection, and the bacterial load will dramatically decrease in the liver of the spleen. I mean, for the OSPC3 deletion strain. The deletion strain essentially is has an attenuated virulence activity. So it turns out that OSPC3 directly targets caspase 4 and 11 and renders an inactive caspase. So through 3 years of biochemical studies, we figured out that OSPC3 indeed is a brand new enzyme. And this enzyme uses NAD as a ligand to modify a specific arginine in caspase 4 and 11 through a modification we call ADP riboxynation, which is a novel post-translational modification, and we actually gave the name. So if you are interested in how to figure out a novel post-translational modification, you might want to read this paper. But for this group of audience, where I will go further with the functional part. So basically, OSPC3 Shigella mutation gave us a tool, a genetic tool that this bacteria now can potently stimulate the caspase 11 Gastamine D pyroptotic pathway. So this mutant bacteria is basically a live attenuated, virulence attenuated bacteria. So in the mouse infection, we see that the mutant bacteria stimulate a lot more anti-Shigella IgG production, as you can see clearly here. In the wild-type mice, if we knock out the caspase 11 or gastamine D, there is no increased antibody production. So after the immunization, we further challenge the mice with a lethal dose of Shigella infection. We see that after infection with the OSP3 deletion strain, the infected mice now have seems to be have a vaccine-like effect, can give a protective effect towards against the second round of infection, and this was not seen in the caspase 11 knockout mice. So Shigella is a bacteria that has no vaccine available. There are two live attenuated strains, ICS-A and Guar-BA. These two strains are currently in development as a live attenuated vaccine strain in clinics. If we compare our OSP3 deletion strain with the two candidate vaccine strain, we see that the Shigella antibody production is even was much higher than the two candidate vaccine strain. If we combine our mutation, OSP3 mutation, on the background of either ICS-A or Guar-BA strain, so we make a double deletion. The double deletion give much higher anti-IgG, Shigella IgG antibody production. And now the immunization or the vaccine-like protection effect was much more dramatic. So we think that the OSP3 deletion stream may give us a way to develop anti-Shigella vaccine. And this study also provide a direct, very strong evidence that the pyroptosis pathway can stimulate antibody production, the humoral immunity, because deletion strain will stimulate the host, the mice, to initiate a pyroptotic response. And this caspase 11 pathway does not process interleukin, so it's a clean, experimental setting. So we not only have gastamine D, we also have gastamine A, B, C, E. There are 5 gastamines in human. The 5 gastamines in human. There are genetic mutations, disease mutation associated with different gastamine family members. So all the family members, they contain this 2 domain, N-terminal pore-forming domain, C-terminal inhibitory domain. The difference is that other gastamines are not cleaved by caspase 1 and 11. They could be cleaved by other proteases. They could be activated by protein-protein interaction or post-translational modification. So we think that the key for pyroptosis is not caspase. The key for pyroptosis is gastamine family of pore-forming protein. Therefore, we redefine the concept of pyroptosis. So what's more interesting is that we not only mammals have gastamines, and recently there are also papers showing that some bacteria also have gastamine-like proteins. And in this paper deposited in the bioarchive, and they show that there are many bacteria, they have gastamine-like proteins, and those proteins are also activated by adjacent protease cleavage. And this locus, the locus gastamine, bacterial gastamine locus is often next to the CRISPR locus. You see the pore-forming activity is very robust. So this pathway also exists in bacteria. You may function in antiphage development. That's a speculation. And in fungi, there are also gastamine-like genes. And in fungi, the gastamine-like genes function in non-self discrimination against genetically incompatible strains. So basically, the fungi have haploids. If the 2 haploids fuse to each other, mate with each other, if the genetic material is not compatible, and then fungal gastamine becomes activated, and then the cells die. So this is the protective mechanism for genetic contamination in the fungi. So for all 5 gastamines in human, we found that 3 of them, ACE, are very difficult to see their expression in cancers. In this study published last year, and we figure out the underlying mechanism. So now I'm switching to the anti-tumor immunity function of pyroptosis. So in this story, basically my collaborator, Dr. Zhibo Liu at Peking University, who is an excellent chemical biologist, he designed a bioorthogonal system that allow us to deliver gasdermin specifically into tumors cells in mice. And he has a small molecule to release the active gasdermin from the internalized nanoparticles. Basically, Dr. Liu give us a tool so that we can deliver and release gasdermin into tumor cells in a selective way in mice. So in this 41 memory carcinoma model, at day 6 and 9 and 12, we induce pyroptosis activation in the tumor, in the mice, in the tumor-bearing mice. After 3 rounds of induction of pyroptosis, the tumor completely shrinks. We see a dramatic tumor regression phenotype, and all other controls works very appropriately. We also see this in other tumor models. The statistics you can see their dramatic response. So you might think that, well, you just kill tumor cells, cancer cells, so you don't see the tumor growth anymore. But that was not correct. This method Dr. Liu designed for us is not that efficient. In the cell culture system, we can only kill about 20–30% of cells through pyroptosis induction, chemical method induced pyroptosis induction. In the mice, we can only kill probably around 10% of cells. So then the entire tumor shrinked, right? So the question is why a few tumor cell pyroptosis could lead to complete tumor regression. So when switch the experiments to a T-cell deficient asymmic mice, we see that with the same 3 rounds of pyroptosis induction, we did not see any tumor regression response. So this data directly strongly suggests that a few tumor cells undergo pyroptosis, and this will trigger inflammatory response, remodulate the tumor microenvironment that eventually cause a T-cell activated T-cell induced anti-tumor immunity. So now I want to remind you that in this very hot tumor immunity field right now, we trace the history of the anti-tumor immunity back to this doctor called William Coley in 1893, and he observed that some severe cancer patients can self-cured without any treatment, and that is, he thinks that is due to the infection by Streptococcus pyogenes. So this is now we call the Coley toxin. So basically, those late-stage cancer patients, they got infected by Streptococcus, and they then recovered spontaneously from cancer. So and that because the bacterial infection triggers the anti-tumor immunity that will give a self-cure effect. So as you might recall at the beginning of my talk, pyroptosis was originally discovered in bacteria-infected cells. So I don't know if there is a connection here, but it's something interesting to further investigate. So we also did some immunological profiling. We see the T cells goes up and the Treg cells comes down. If you deplete CD4 and if you deplete the CD4 or CD8 positive T cells, you can reverse the phenotype. So that's sort of expected thing. And with this experiment, if we reduce the 3 rounds of pyroptosis induction into 1 round, and this effect is not sufficient. With 1 round of pyroptosis induction, we did not see any tumor shrinkage phenotype. And this 41 tumor model is very well known to be a cold tumor. It also resists the checkpoint blockade therapy, and the PD-1 antibodies treatment did not cause tumor shrinkage. However, after 1 round of pyroptosis induction, and we follow that by PD-1 antibody treatment, we see a dramatic tumor shrinkage phenotype. So this suggests that a little bit of pyroptosis induction within the tumor, in tumor cells, and that sort of serving as a flame to convert the cold tumor into hot tumor. So that also confirms the anti-tumor immune function of pyroptosis induction. And this also explains that why cancer cells want to silence gasdermin expression if they could. because if they keep gastromin expression, if gastromin gets activated, by some accidental event, and then the tumor will not grow because it will trigger a tumor immunity. And lastly, I want to share you with another story. In this story, basically we show that gastromin-mediated pyroptosis also play a role in a normal endotumor immune process, because the previous experiments, we use an artificial way. The chemical biological system allow us to deliver gastromin into tumor cells. So in this assay, the question we want to ask is, in the normal endotumor immune process, the T cell or NK cell mediated killing of tumor cells, is there a pyroptosis involved? So the CTL, cytotoxic T cells, they deliver granzymes, 5 granzymes into target cells. The activity of those 5 granzymes eventually will lead to the killing, the cell death of target cells. And the granzymes are serine protease. There are also 5 granzymes in human. And the textbook tells that the granzymes kill target cells through apoptosis. Is this always true? So in these experiments, we use the model target cells, 293 T cells. And the 293 T cells is green. We load it with a green fluorescence. And this dye cannot get out of the cells unless the cells undergo lytic death, like a pyroptosis. So we use NK cells to attack the small NK cells to attack 293 T cells. And you see that in normal 293 T cells, we did not see a leakage of the green fluorescence, this dye, suggesting that there's no lytic death here. However, 293 T cells does not express any gastromin. So we just put 5 gastomins each into 293 T cells, generate stable cell line. And then when we express gastromin B in the cells, when the NK cells attack 293 T cells, you see the green fluorescence will leak out instantly. You also see the propylene iodide, the DNA dye, goes into the 293 T cells. So this suggests that the presence of gastromin B can switch the killing by CTL or NK cells from apoptosis to pyroptosis. So if we do Western blot, we see that granzyme A, there are 5 granzymes, right? Specifically, granzyme A, the serine protease, can cleave gastromin B also within the linker region on lysine residues. You can see the 2 domains, N-terminal pore-forming domain, C-terminal inhibitory domain. The cleavage, of course, will release the N-terminal domain from pores and then execute a target cell pyroptosis. So this gene, gastromin B, another gastromin, small bomb, right? The 5 gastomins. This gene is highly expressed in the digestive tract system. If we take cancer cells from the digestive tract system, like colorectal cancer or esophagus cancer, we give the cells granzyme A, you see all the cells undergo pyroptosis. There are big bubbles almost from each cells. You see the very robust cleavage of gastromin B upon delivery of granzyme A. And for CAR T cells, CAR T is artificial engineered cytotoxic T cells. When CAR T cells kill target CD19 positive cells, if the cells express gastromin B, and then gastromin B gets cleaved, right? And the cells undergo pyroptosis. And if you make a mutation to make the mutation in the cleavage site to block the cleavage, you also block the pyroptosis. If you knock down granzyme A in CAR T cells, you can also reduce the pyroptotic killing. The same is true with TCR T cells, because TCR T is another cancer immunotherapy that is under development. Is very similar to CAR T cells. So just to summarize that, in normal anti-tumor immune process, and the CTL would generate interferon gamma. Very interesting, interferon gamma will stimulate the transcriptional induction of gastromin B. In the right side, you see that various cancer cells, you don't see gastromin B expression, but if you treat them with interferon gamma, and sometimes TNF alpha, you see a dramatic transcriptional induction of gastromin B. And then granzyme A delivered into the cancer cells by CTL will cleave gastromin B and then release the N-terminal pore-forming domain. And it will make pores on the plasma membrane. The target cancer cells will undergo pyroptosis, and pyroptosis will trigger a strong inflammatory response. And this will further remodulate the tumor immune microenvironment and further stimulate the cancer immune activity. So we can see that the gastromin B pathway also give a synergistic effect with anti-PD-1 checkpoint therapy in this CD26 mouse syngeneic model. So basically, those gastroamines like B and E could serve as a biomarker for cancer immunotherapy, because if the target cells express gastroamine, they probably are more easy to respond to, more likely to respond to the checkpoint blockade therapy. You could develop agonists for gastroamines that might be the next generation cancer immunotherapy drugs. So I want to thank the students and postdoc in the lab who did the work. Originally, gastromin D was identified by Jianjin Shi and Yue Zhao, and I did not talk about the gastromin E work. And the ASP-C3 work was done by the postdoc fellow Zilin Li. And the last piece of work on granzyme A cleavage of gastromin B was done by Zhiwei Zhou and Hua Bing in the lab. And I want to thank Fengchao Wang who generated knockout mice for us. I'll stop here. Thank you.
Thank you, Dr. Feng Shao. So is there any questions?
Hi, Dr. Shao. It's very inspiring talk. So I have one question. You mentioned that it's amazing that bacteria can trigger this. Not bacteria. I mean, pyroptosis actually can trigger anti-tumor immunity. And you mentioned that cholestoxin actually 100 years ago that you can use it for tumor immunity. Actually, they also use the heat inactivated bacteria in this condition. And it seems this kind of pyroptosis may not be directly induced by this bacteria, or there's an indirect way. So I'm wondering whether you have any opinion on this. Maybe it's indirect through the immune system.
Yeah, so they could use the inactivated bacteria, but the inactivated bacteria still contain lots of PAMs, like LPS, flagellin, and those PAMs can still stimulate pyroptosis. So, so an induction of pyroptosis by bacteria do not require live bacteria. You could give the macrophage or other cells some bacterial toxins, and the cells can still undergo pyroptosis. Yeah, there's that could be an explanation. Yeah.
They can do intratumor injection of this toxin. Then they can also do like a IV injection, and they show that both of them can work. So if it's an IV injection, they could work on other cells. How does it trigger this specificity?
We don't know. That's a good point. So we're now trying to replicate we're now trying to model this in the mice. So because it was original observation was in patients, right? And I don't think anyone actually followed up on that using like mouse model or other animal models. So we're trying to model that, whether we see Streptococcus infected mice indeed have a stronger anti-tumor immunity. Yeah.
Right. And you actually just show that T cell can trigger this pyroptosis in the cancer cells, right? So that's why I'm wondering whether there's an indirect, the heat inactivated or some live bacteria can work through some immune cells and then trigger this pyroptosis.
Yeah, so very could be very. So I think the pyroptosis probably occurred in either macrophages or some epithelial cells.
Yeah.
And that then signals through complicated immunological process to eventually activate the T cell mediated immune response. Yeah.
All right. Thank you.
Yeah. So, but if you think about the this bacterial experiments, basically, the mutant bacteria can generate a much higher antibody production. So basically, the mutant bacteria compared with the wild type bacteria, the mutant bacteria can induce pyroptosis, right? So this data also strongly directly demonstrate that the whole cell pyroptosis in response to bacterial infection can promote antigen presentation or antibody production. Yeah.
So I didn't have a chance to read your Nature paper yet. So this bacterial effector, the modification of the arginine, how does it inhibit the activity of it was caspase, right?
Yes, it modifies a specific arginine, and that arginine actually coordinates the aspartic P1 site in the substrate. So that's the most key arginine, key residue in all caspase. So caspase cleave after aspartic acid. The aspartic acid in the substrate is coordinated by an arginine in the caspase, and that arginine is modified by ASP-C3.
Dr. Shao, fantastic talk as usual. So I just curious about whether cancer cells have any resistance mechanism to gastromin pathway. So there's many mechanisms tumor cell can come up to enter, like pharoptosis, apoptosis, whether any specific mechanism to enter, pyroptosis.
I think the major mechanism in cancer cells is the epigenetic silencing. As I mentioned that if we have measured like 100 different cancer cell lines as well as some primary tumors, so you can see gastromin D expression ubiquitously, almost ubiquitously. But other gastomins like A, C, E, and I would say majority of the cancer cells have silenced the expression of gastromin A, C, E, and I think that's the major mechanism. So the reason that gastromin D was not silenced, probably because gastromin D is too important for innate immunity, right? Because all the inflammations, canonical, non-canonical inflammations, they all converge onto gastromin D. So, but other gastomins, they have tissue-specific expression, and then I think basically tumorigenesis is an evolution process. If they did not silence gastromin expression, they could, if they get activated, it could trigger strong anti-tumor immune response. But again, there could be other mechanisms for cancer cells to avoid the activation of those pathways. I think there will be more studies coming out in the next few years.
Okay, Shu has a last question. Okay.
Beautiful work. I have a question. During physiological processes, such as antibacterial infection or anti-tumor immunity, do you think there will be multiple forms of cell death, including apoptosis, pyroptosis, and necroptosis at the same time? Or there will be a dominant form of cell death? If this is the case, do you think if each form of cell death would inhibit each other? Yeah, thank you.
Very good question. So in my talk, basically we are in an experimental setting. We make the system is clean, apoptosis is clean, robust pyroptosis. I think you're absolutely right that in real world, very often is a mixture. Probably is a mixture of apoptosis and pyroptosis. But for a mixture form of cell death, I think the biological consequence is determined by PS exposure and membrane lysis, which one occurs first. If PS exposure flipping occurs before gastromin pore formation, and then you got an apoptotic response. That's the cells, dying cells, just engulfed by macrophages. But if a PS exposure occurs after gastromin to make pores, and then you leak out the cytosolic contents. So that's my experimentation. We also have another piece of work that is under development if we manipulate the system, the target cells into pure, clean apoptotic death, in an experimental setting, when they encounter the CTL, indeed apoptosis inhibits anti-tumor immunity.
Okay, so let's please join me to welcome. Thanks, Dr. Feng Shao, for the wonderful talk. Thank you.
Thank you. Thank you.
So let's move on. So next speaker is Dr. Baoxue Ge. So Dr. Ge is a professor of the Tongji University. His expertise, he's expert of the TB and the host and the pathogen interactions. Mostly he works on the TB and how does the TB interact with the host. So let's welcome Dr. Ge. Thank you.
Thanks, organizing committee to invite me to give a talk. It's a great pleasure to share with you my work. So today I am going to talk about innate immune molecules, but I switch mostly to the TB research. So most of the talk will about tuberculosis. It's a very important bacteria. So all you know, the virus. Actually, I started my career with virus research when I was in Pasteur Institute, Shanghai. Actually, 10 years ago, I moved to TB. Either, the bacteria or virus, they will trigger the innate immunity. So the first thing is, so I know skip this because I'm after the talk by the Feng Shao. So today we are focused actually my research is 3 points. So actually the first one have been well studied, we have published a bunch of papers. But the second is the key point is TB, how the tuberculosis, the interaction between the TB and the innate immune system. The third question is if or whether innate molecules have other function other than, the innate immunity. Each of question we have done some work about it, but today I will tell you some story about question 2 and question 3. So as all you know, TB is a big problem now in the worldwide. So there are 3 major kinds of infectious diseases in China. One is hepatitis virus. Another one is HIV. The third one is a bacteria. Most of infectious diseases are virus. Most now in the COVID-19. But the most TB have killed many people. This is a summary. So every year, WHO, World Health Organization, will give a report about TB. So this year, 2021, we give a report of last year, it's 2020. There are 2, I think we should pay much more attention. The first one is a death toll. So there are many more people died than before, since 2005. And the single case for TB is about 1,028,000 people. And then HIV, together with TB, there is another 200,000 people. So together we have 1.5 million people die. So that's an increase. And WHO expected the TB deaths going down every year to the 2050. We are going to end the TB. That's our goal. But now the goal, we don't have the chance. Okay, because it don't decrease the increase. The second thing is now China is the number 2, and number 1 is India. So I think several years before, we are like rank 3 or 4th, but we moved to the 2nd place. That means we have more TB infection now in China. So in clinical trial, in clinical case, the TB, the difficult part for clinician is drug-resistant strain. So only 50% resistant patient can be cured. So now a new drug, beta-quinine, moved to the hospital. So now we can get to the 8.8%. But we expect, I just read a report this morning, we are going to expect the drug resistance too for beta-quinine, because beta-quinine also targeted the bacteria. So I think for China, there's another big issue is we are getting more big population of diabetes. Diabetes will increase the risk of TB. Yeah, we already have the evidence. You have high level sugar, the glucose. The bacteria cannot be cleared. So all of this are big challenges for TB research. But we don't see many people doing research in TB because this requires the biosafety lab. So my research focuses on the mechanism. The mechanism is, every year, in the campus, there are people, especially for young people, they will be active disease. So for the immunity to TB, because we always know that's a lung problem, right? So I'm from a lung hospital. So it's the respiratory tract disease. So first, this bacteria is intracellular bacteria. They get into the macrophage, and the macrophage will go to death. So that's a mixture, okay? We already have some evidence to show it's a mixture: pyroptosis, apoptosis, necrosis, ferroptosis. So we're still trying to figure out how it's regulated. So normally the apoptotic macrophage will engulfed by DC. Then DC will move to the lymph nodes to present the antigen to T cells and differentiate the Th1. Th1 go back to the lung, will secrete interferon gamma. So that's a second killing. The interferon gamma will activate macrophage to get rid of the bacteria. So that's a normal route. So for the vaccine development, mostly is focused on how to increase the number and function of Th1 cells, the interferon gamma T cells. But the problem is still we have toxic strain. Yeah, everybody in the room, you probably have been curated by BCG, right? So that's a Chinese name Ka Jiemiao, right? That's a attenuated strain. The toxic is not so toxic, right? Some bacteria like H37IA, it's non-toxic. So this toxic and non-toxic mean what? The non-toxic strain can be easily get rid of by our immune system. For some toxic strain like H37RV and a Beijing strain, now the most epidemic strain is Beijing strain. You see, we're trying to get rid of the COVID-19 to be Chinese virus, but we get this name, Beijing strain. Okay, that's the TB. So how can this strain be epidemic? Because it inhibits our immune system. So we switch apoptosis to necrosis to that way
the bacteria can spread. So we are now, with the help of Feng Shao, we are also studying from the paraptosis, right? So how they switch to be, protective or to be toxic. Then get rid of, to become a granuloma. The bacteria will be inside. So I think 10 years ago, I started the TB research. I'm not focusing on the interaction between the bacteria and the immune system. In other words, we are trying to identify the toxic factors from the bacteria, and then to see how these toxic factors to interact with our immune system. Actually, it's how they inhibit our immune system, right? To get to spread the bacteria. We have published a bunch of papers. So actually, the bacteria is a little bit different from virus. So they had everything, right? The TB, they had like 4,000 proteins. These proteins, some are on the cell wall, some can be segregated. That means they get the chance to interact our immune system. There's a list of a review paper that lists a bunch of proteins. They can either activate, our innate immunity or inhibit innate immunity. So, in addition to cell wall component, they also have a secreting system. They are very strong. The secreting system. Some other bacteria also have it, but this TB is very strange. They can secrete about like 300 secreted proteins. What do these secreted proteins do? So we have cloned 208 MTB proteins, and they're doing a screening. You can see some proteins activate NF-κB pathway, but some other proteins inhibit the NF-κB pathway. I still don't understand why they activate and also inhibit, but overall, the bacteria is very mild, very mild. They don't have fever, okay? If the patient are infected with TB, they don't have the fever, the high level fever. So very mild, okay, the immune system. So that is keeping the balance. So that's one protein we already published, MT53. So just for cutting short, it have SS link. They can be interacted with TAC1 to activate the NF-κB pathway to induce the cytokine production. So actually, this component we can be used for the vaccine development. So another protein is inhibited our immune system. Actually, it's metabolite inline. So basically, this when TB infects inside the cells, it will secrete RV0222, but it's inactive. It needs to be activated by our ubiquitin system. That's the E3 ligase, NAPC2, to get ubiquitinated. That's a K11 ubiquitinated, then get activated. That means for the TB infection, our host also be involved. Nobody is innocent, okay? We are also guilty. To be help RV0222 to enhance SAGP1 binding to the TREP6 to inhibit our immune system. So as I said before, the cell wall of TB is very thick. It's very, unique. They have the PGM, but they also have some lipids and the AG. That's sugar. So already, because for TB now we have a bunch of drugs, okay, to anti-TB drugs. But originally find out that EMB can target the biosynthesis of AG, but nobody knows the mechanism. But then last couple of years ago, Professor Saw, they distributed the structure to find out the AG actually binding the structure. So for the AG, it's very hard to get purified. So you can not do much research about AG. But Professor Ye, they have synthesized this AG, and also they published this their research. Okay, then we easier for us to get the AG to look at the immune function of AG. Because original AG is just a component of cell wall, so you can develop a drug targeting the AG biosynthesis. But people are still been working on it to trying to block the biosynthesis of AG, and that way you can kill the bacteria. So actually we made a pattern for AG. You could block the AG, actually the lung damage going down. You can inhibit the lung damage. So actually AG is not only the component of cell wall, it can also is PMPAMS. It can induce expression of MMPs. And what's the receptor for the AG? Because it's a sugar, you can binding with the galactins. So we did the bunch assay, find the AG actually binding mostly with galactin line. So if you lock out the galactin line, then AG cannot induce MEMS4. Also, we did the animal infection, this is into vascular injection of AG. So we summarize with is AG can have an immunological function, can go through a galactin line to activate ERK, and then to induce MMP to cause lung damage. The TB infection is very complicated. So it can, as said before, it can form granuloma. So there is active TB. There are also latent. Actually, the whole world population is 1/4 people are infected by TB. But they are latent TB infection. So when you getting old, you can be reactivated as a recurrent or the disease. So for the granuloma, it's a little bit similar to the tumor. So in the inside region, there is a bacteria, it's a macrophage, it's hydrophobic. And then circle around is T cell and B cells. And then the outer is the fibroblast to form a granuloma, the bacteria inside. But when you, the immunity is lower, the granuloma will be activated to release the TB, to become active disease. So how the bacteria to coordinate hypoxia and their anti-immunity function? So we did an assay to find hypoxia can induce a protein, a bunch of proteins. We focus FDA, this protein is also called RV0589. So actually it's acetyltransferase. So this protein, if you knock this gene from TB, then you get more expression of IL-6 and other inflammatory cytokines. So we did because normally this expression of IL-6 is controlled by the MAP kinase, but MAP kinase inhibitor don't have any effect. So then we suspect that maybe it's epigenetic histone modification. So actually we use histone inhibitor can block the expression of IL-6. And also it shows the FDA actually suppress IL-6 through the HK9 acetylation. There is acetylcholine A is substrate for the histone acetylation. So I say FDA actually is the acetylcholine acid transferase. It can synthesize 2 acetylcholine A to the acetylcholine. That way it can decrease acetylcholine level. If you mutate the enzyme site, it cannot work anymore. Yeah, this is my favorite. So for the granuloma, if you take a picture, in the hospital, you can see your lung have a big spot, right? But for the mouse, the C57, it actually is anti-TB, okay? They cannot form the typical granuloma. We use this is a zebrafish model. Okay, we use a zebrafish to infect it with marine. You can see the very typical granuloma, and the bacteria is inside. So you can see 2. One is if you lock out IL-6, then you get more granuloma and more bacteria. The second is FDA functions through IL-6, right? So if you knock out FDA, you also can get more bacteria and granuloma. So to summarize this finding is actually there's another component, the AC6. They can going through the TCA cycle to produce more acetylcholine A. Then they go through the histone modification to synthesize, express IL-6 to inhibit the granuloma. But for the TB, especially under the hypoxia, it will induce another viral toxic factor, virulence factor is FDA, that it can synthesize 2 acetylcholine A to the acetylcholine. Then the acetylcholine A, the level going down. Then that way it can to promote the formation of granuloma and the bacterial growth. So last few minutes, I will talk about the C-gas. A C-gas is a skin sensor that's already, 2013 by Zhijian Chen found. Actually, it can synthesize even virus, bacteria, and our own DNA. So C-gas not only responds to the virus infection, double-stranded DNA, also can link the genome instability to innate immunity. It will form the micronucleus. The micronucleus will easy to rupture and then release the DNA, will activate the C-gas to activate the interferon pathway. But the question is C-gas also involved in the maintenance of genomic stability? So we find actually the C-gas involved in DNA damage and repair. The C-gas is normally in the cytoplasm, but when you treat it with a genotoxic stress, you move to the nucleus, the binding with timeless, and then binding with the PAP1 get rid of timeless to block the DNA repair pathway to increase the genome instability to cause the tumor. So this research identified another function for C-gas. And for the evolution view, even more earlier than the function of C-gas as an antiviral factor. Then we started being interested in the micronucleus. Actually, the Bounty reference have mentioned in many diseases you can see more accumulation of micronucleus. But exactly what's the function of micronucleus is unknown, still unknown, because you have to get rid of it, right? So the question is how this micronucleus to be cleared. Very small, you can see that below, right? For the autophagy receptor is the ARR, the ALC3 inactivation is a very important marker. interestingly, we find that the C gas have this AR motif, but exactly the C gas is autophagy receptor, right? So we also this very conservative. So we find C gas actually can interact with ALC3 in the micronucleus. You can see the right side, right? And then can deliver the autophagic micronucleus to the lysosome. To summarize this, C gas is a micronucleophage receptor. It will be required for the clearance of micronucleus. So during the replication, chromosome replication, it can form the micronucleus. Then C gas can bind with micronucleus and direct it to the lysosome to get rid of micronucleus. And otherwise, micronucleus will induce the interferon, the innate immunity. Okay, I will thank my colleagues and NSFC and Ministry of Science and Technology. Thank you.
Is there any questions?
Yeah, this micronuclei targeted to the lysosome. I thought, does the lysosome contain also nucleases that can degrade the DNA? Or I thought lysosomes only contain proteases that can degrade proteins. But what happens to the DNA that's contained in the micronuclei?
Oh, they also in the lysosome have the DNS.
Their DNA.
DNS, they will digest the DNA. Yeah. There's another paper published in the Nature 2017. Their group is mostly working on the micronucleus. They have checked the DNS and RNS in also you can digest the.
Okay.
Yeah.
I noticed that in a lot of the micronuclei actually doesn't have the. It's not targeted to the autophagosome, right? Or the lysosome. So, is only a subset of the micronuclei that's targeted by this pathway, or is all of the micronuclei can be targeted?
Not all of them. Yeah, you are right. So only the percentage is very low. Percentage is very low. Yeah, 10 or 15%. So that's a question. Yeah.
Yeah, I have a quick question. So, I guess how does C gas get into the nucleus? So, do they have the nuclear localization sequence?
Yes, they have two. They have two signals. Okay. The AIS2, but only two have the infector. If you mutate two, then they cannot get in the nucleus. Because at the time I didn't show the data.
I see. Cool. Okay. So, are there any questions from the students, trainees, or whatever postdocs? We didn't hear any question from you all, okay? Feel free to ask any questions, okay? Okay, if you have no more questions, let's thank you, Dr. Ge. Thank you for your wonderful talk. Okay, so let's move on. So last speaker is my previous PhD supervisor. So I'm familiar with him very well. So Dr. Jianmin Zhou, he's from Institute of Genetics and Developmental Biology case. So Jianmin got his bachelor from the Sichuan University, and then he moved to US, got his PhD, and he did postdoc training also in the same university, Purdue University, and then he moved to the Kansas State University and has his own lab for study the plant and bacterial interactions. So early on, he studied a lot of story which is called long host resistance of from the plants, and then he moved back to NIPS in 2004, I guess. And he started his own lab at NIPS, and I was really lucky to get into his lab and get a training when I was the graduate student. And early on at NIPS, he studied a lot of study about the bacteria, which is cause the disease in plants. Actually, like the animal, plant has the innate immune system as well. And his study was focused on how the bacteria effect, like Feng Shao did in mammals, and how does the bacteria effects can hijack the plant innate immune system. And then he discovered really cool stories and published a lot of papers as well. And one was really amazing work he did is about the Zasmolos, which is the bacteria caused the disease in plants as well. And he discovered AVR-AC, which is has a really cool modification, which is a unique modification. And with this story, he get into the really long-term having studied biological line between the plant and the host and the pathogen interactions. Actually, so bacterial infection causes the plant called the hyper reactivation, which is called HR. So that means the plant will be die soon, very early and very quickly. So, and finally he got is which is called a program cell death as well in plant. And now his work uncovered all this, the mechanism underlying this phenomena. So let's welcome Dr. Jianmin Zhou, and his title, which is called a plant surveillance network monitoring bacterial virulence proteins. Let's welcome him.
Thank you very much, Zixi. Got your advisor's record straight. I also like to thank the meeting organizers, particularly Dan Shen, for putting me the only plant speaker into the program, which is really unusual. Let me share my slide, share the screen. Okay. Just a sec. Okay, I hope you can see see my slides. So as Zixi just mentioned, we study plant immunity. So as the only plant speaker, I think I'm obligated to give a little bit of introduction about the plant immune system. I try to keep it short. So plants have innate immune system, but does not have specialized immune cells, has no adaptive immunity. And the first batches of immune receptors or innate immune receptors actually were discovered in the 1990s in both animals and plants about the same time. Actually, plants, I think, found the receptors slightly earlier than the toll-like receptors. Okay, so I don't have to give this audience about introduction of the immune receptors in animals. Basically, they can be broadly classified as surface immune receptors and intracellular immune receptors. Plants' immune receptors also have a surface immune receptor and intracellular receptors. So what's striking is that the surface immune receptors in plants are somewhat analogous to toll-like receptors in animals, whereas the intracellular receptors in plants are exclusively similar or homologous to the animal not-like receptors or nucleotide binding oligomerization domain receptors. Okay. So in animals, what both the surface receptors and the intracellular receptors, they perceive rather conserved microbial patterns or PAMPs. In plants, the surface immune receptors, which basically receptor kinases or receptor-like proteins, they perceive conserved microbial patterns, whereas the not-like receptors or NLRs, they perceive pathogen effectors that are delivered into the host cell. Okay, so that's a distinction from the animal not-like receptors. They don't perceive conserved patterns. They perceive virulence factors delivered from pathogen into the host cell. Okay. So as Zixi just mentioned, my lab has been studying plant interaction with bacterial pathogens, including Pseudomonas stringi and Xanthomonas campestris. This bacterial pathogen can cause nasty diseases on many different plants, including crop plants. Let me show you some of the disease over here. Okay. This bacteria actually use the so-called type 3 system to deliver a variety of the effector proteins into the plant cell. Okay. The subjects of concerns are the plant immune receptors, including surface receptors and the intracellular receptors, which come in hundreds in each plant species. Okay. So our question is trying to understand how plant immune receptors work and whether we can apply some of the receptor genes or resistance genes in crop plants for disease control. So a number of years ago, a former graduate student in my lab, Zhang Jie, he was studying bacterial effector protein for the AVR-PPHB, which is the carried by the bacterial pathogen Pseudomonas stringi. And that once enter into the host cell, this protein actually cleaves a protein kinase called the BIC1, which is cytoplasmic kinase. Okay. And this work actually was also involved a lot of work from Zixu. He's an important contributor from the work as well. So what's important about this BIC1 is that it is a central player that operates under downstream of a number of surface receptors, including the flagellin receptors and the elongation factor 2 receptor, which is bacterial protein, and also receptors for fungal cell wall components typing, which is with polysaccharides. Okay, they all operate through BIC1 or its homologous protein PBLs. Okay, to trigger downstream immune responses. So this effector protein AVR-PPHB can cut this BIC1 protein and to destroy this pathway to block the immune activation. So over the years, in my lab and other labs in the world has figured out that how BIC1 trigger downstream responses, including activation of MAP kinase cascade, activation of reactive oxygen species through the plasma membrane NOx protein or ultraviolet G protein, etc. And also calcium channels. In 2012, I think this Zixu also mentioned this, that we're studying another effector protein. This is called AVR-AC, is carried by a bacterial pathogen Xanthomonas campestris, which is a pathogen caused black rot disease on cabbage. Yeah. So this turns out this protein is a unique enzyme that uridylates BIC1 protein. It basically adds UMP to the key residues of BIC1 to block the activation of the BIC1 kinase, thereby block the immune signaling. Okay. Later, another student in my lab, Wang Guoxun, he discovered that another protein called PBL2, which is homologous to BIC1, it can be similarly modified by AVR-AC. But this modification does not block plant immunity. Instead, this modified protein recruits a protein complex, including ZAR1 protein, which is a typical NAR receptor, and another pseudokinase called RKS1. So this forms a tripartite protein complex. Once this is formed, this triggers very strong immune response and cell death. So as I mentioned earlier, that the plant NARs and the animal NARs are very similar in its architecture. Okay, so the most striking feature are the central nucleotide binding oligomerization domain or NOD domain, okay, which in animal has been shown to mediate oligomerization to form inflammasomes triggering immune responses. And this C-terminal leucine-rich repeat domain is responsible for ligand binding. In plants, these NRs also contain these two domains, but the N-terminal domain is more variable in plants, including TEER domain, COI domain, and RPWA domain. Okay, so therefore they are called RNL, CNL, TNL, etc. Okay. In animal, this the N-terminal domain is also highly variable. So a key question, I think since the identification of the first set of plant immune receptors, that whether any of these NAR proteins in plants will be activated similarly, just like the animal NAR proteins. So that is whether they also oligomerize to activate immunity. So my graduate student, Wei Juan, he did the deoxygenation assay to show that the before activation, the ZAR1 protein actually stays as monomer. But upon activation, the ZAR1 protein shifts to large molecular weight, which indicates is oligomerized, which she can further show to be the case by using the blue native gel assay. Later, we collaborate with Cai Jijie's group, and his group showed that these proteins, these three proteins, can be assembled or reconstituted in vitro into a large complex. Okay, and when you use cryo-EM, you can see this complex actually is oligomer. It's a pentameric complex. So you have three proteins here: is PBL2, the modified form RPS1. Here down in the middle is the ZAR1. So you have 5 of them together form this complex. So we call this resistosome. Basically in parallel to inflammasome in animals. So what's striking from the cryo-EM structure is that this ZAR1 resistosome, the N-terminus of the CC domain actually form a funnel-shaped structure. There's a side view, this is top view. As you can see here, there's a small hole in the middle that's about 6 to 7 Armstrong. It's very small. But the outside of the funnel-shaped structure is hydrophobic, whereas the interior is hydrophilic. So this is suggestive that it could be associated with the membrane. And we did the membrane fractionation assay. We can show that yes, indeed, the ZAR1 protein upon activation can be enriched in the plasma membrane. So this is suggesting that the ZAR1 resistosome can form a pore in the membrane. So ZAR1 really is the first resistosome shown in plants. But later, I think last year, Cai Jijie's group discovered another resistosome, which is a typical TNL, the N-terminus of which is TEER domain. Okay, you can see here the TEER actually is clustered in the middle, but in this case, the TEER domain does not have a pore forming function. Instead, TEER is an enzyme called NADS, which cleaves NAD into a variety of derivatives, which could function as single molecules. So the oligomerization of the TEER serve as a way to activate the NADS. Okay. So this is a comparison with the animal NRC4 inflammasome. You can see the oligomerization is similar to the plant resistosome. Okay. So as you have all heard Professor Cao Feng's talk, and in the animal inflammasome, a major function of which is to recruit caspase to activate gastramine D and also other gastramines as well to form a pore in the oligomer pore in the membrane to trigger pyroptosis. Whereas we suggest that ZAR1 resistosome could also associate with membrane to form a pore, but this pore must be small. And this can trigger cell death, which we call hypersensitive cell death. Okay. So there's really a number of questions not answered from the prior structure study. That is, whether the ZAR1 resistosome truly form a pore in the plasma membrane, which is something we want to explore. And also, What is the function of this pore? Since this pore is very different from the gastromin D pore. So we collaborate with two other PI at IGDB, Professor Chen Yihang and also Professor He Kangmin, try to tackle the questions whether ZAR1 resistance truly form a pore in the plasma membrane, and what is function of this pore? Is it an ion channel or something else? And how does it kill the cell? So we first use the single molecule imaging, trying to follow the ZAR1 protein at the plasma membrane to see whether it's truly oligomerized in the plasma membrane or not. As you can see here, this is a GFP tag ZAR1 protein activated form. You can see it forms spots, aggregated spots in the plasma membrane, and it moves in the plasma membrane, just like as other plasma membrane resident proteins. And if you calculate the intensity of the GFP of each spot, you will see that the average intensity is about 5 times of the monomeric GFP. So this indicates this in plant cell, ZAR1 resistance also form in the plasma membrane as a pentamer. So we next try to ask whether ZAR1 resistance has channel activity. So this first experiment is done in the Xenopus oocyte. So we reconstituted this ZAR1 resistance by expressing the 3 components, plant components, and 1 bacterial component. As you can see, this activated ZAR1 can indeed trigger the current traces in the oocyte. Very beautiful current traces here. And note here, in the interior cavity of this ZAR1 resistance, the presumed channel cavity, there is a key residue at the 11th position, glutamate, which caught our attention because this residue is conserved among this group of protein, and also this residue is required for its immune function and also cell death triggering function. If we mutate this residue, substitute into a non-charged residue, you can see the current traces basically are largely diminished. But if you substitute with another acidic residue, aspartate, then this current traces is retained. So this tells us that ZAR1 resistance is indeed active channel. Okay, and this 11th position of glutamate is essential for its channel activity, and likely caused by this carboxylate ring formed by glutamate 11. So we decided to further characterize this channel property by using lipid bilayer experiments. Here in this device, you have the synthetic lipid bilayer. On this cis chamber, you can add the pre-assembled resistance in it. And this readily allows this resistance to insert into the membrane. And in the 2 chambers, you can add different buffers. Then you can add the voltage to record the current traces. So this you're looking at here is the single channel trace. You can see on both sides, both positive and negative voltage applied. And then you can change the buffer in the trans chamber into either sodium gluconate or TEA chloride. Okay, this gluconate is a large ion, does not pass any known channels. Okay, so this does not affect the channel activity on both sides. But when you have the TEA, which is the large cation, does not pass any channel, this will block the current traces on this side. So this indicates that this ZAR1 resistance is a cation selective ion channel. So we further compared different cations, including sodium, potassium, cesium, magnesium, and calcium. As you can see, that potassium is similar to sodium, so there's no selectivity between these two. And also the cesium is less permeable than sodium. Magnesium is about the same as sodium, whereas calcium is slightly better than sodium. So the conclusion of this is that the XAR1 channel is permeable to calcium, but it's not highly selective. But this is somewhat similar to reported calcium channels in plants, which are all cation permeable channels, not selective. So we decide to ask whether this activation of XAR1 channel in the plant cell can trigger a calcium flux, because calcium is known to be a critical early signal in plant immune responses. As you can see here, the activation of XAR1 can trigger a strong calcium influx. But if you mutate the glutamate 11 position, then this mutant form has very weak calcium influx. So this indicates the channel activity is important for the calcium influx triggered by the XAR1 resistosome. And you can do this in the entire plants. So this is showing the calcium flux upon the bacterial pathogen infection. You can see strong calcium influx, where the mutated form is basically largely diminished in this calcium flux. And we can do similar experiment by by adding the calcium channel blockers. You can also block the immune responses and the cell death. So the conclusion is that the XAR1 resistosome actually indeed form a pore in the plasma membrane, and it is a channel that cation permeable channel that also allow calcium to enter into plant cell, as calcium concentration outside the cell is known to be very high, inside the cell is very low. Okay. This is essential to trigger downstream immunity. As I mentioned earlier, that CNL is just one class of the NAR proteins in plants. What about other classes? So recently, Jeff Dango's group showed that another class of NAR, called RNLs, also can oligomerize and permeable to cations, including calcium. Okay. So their conclusion is that this RNLs also function as a calcium channel to trigger immune responses. And it is priorly known that the third class, or TNLs, actually function upstream of RNLs to trigger immune responses. So RNLs they are the calcium channels. So this basically provides us a unified view of the plant NAR proteins that they probably all function through this calcium channel mediated function to trigger downstream immunity. So calcium really is the first trigger or signal in immune activation. So what about cell death? So I'm going to show you the plant cell death, by using live cell imaging. So this is the infected cell. Here is you're looking at the living mesophyll cell. At the later stage, you'll see the plasma membrane damage, and immediately after that, the cell collapse. So this is a really abrupt collapse, very quick. And this cell has cell wall, so we decide to remove the cell wall and follow the death of protoplasts to see whether we can see any features similar to the animal cell death, paraptosis, etc. So here you're looking at this cell, the membrane damaged immediately after that, the cell collapse. But during the entire process, we don't see any cell swelling, and also we did not see any unique structure like the parotic bodies. And down here, you're looking at the vacuoles. This is this DFT labeled vacuoles. You'll see that hours before the cell collapse, the vacuoles of the dying cell is already abnormal. Okay, this one. So what this tells us is that the cell, before the cell death, there are a lot of things happen to the cells. Okay, so the membrane pore formation itself is not sufficient for to cause cell death. You know, it involves a lot of downstream processes which we don't know. We therefore monitored another marker, which is accumulation of hydrogen peroxide in the cell. Okay, so this is a hallmark of another hallmark of plant immune response, just like in animal immunity. So here we're basically tracking two cells using the dye. You can see the hydrogen peroxide basically accumulates over time for several hours until the last moment the plasma membrane basically leaks and the cell collapse. Here's another cell shows the same thing. Here's the plot of the two cells. If you plot all the dying cells, you're basically seeing the same thing. The hydrogen peroxide accumulates over continuously until cell collapse. So we still don't have the final answer how the cells are killed. But what we can do is we can put these events, cellular events, all together. You can see that the calcium influx and the accumulation of hydrogen peroxide is happen very early. Then after that, you have the organelle abnormalities. Okay, only in the last moment, the last 5 minutes, you'll see the plasma membrane damage, and then followed by abrupt cell lysis. Okay. So we're very interested in how calcium is causing all these different downstream events and what finally kills the cell. So in the last 2 minutes, I'd like to bring attention to ZAR1 protein by saying that this is actually among all the plant NLRs is very unique in that it forms complex with two family of kinases. Here is pseudokinase family. Here's another PBL kinase family. So the many members of it can form different combinations. So these different combinations in the form complex with ZAR1 allow this protein to detect multiple pathogen effector proteins. So this is really form a large network to sense different pathogen effectors. So according to a study last year, it shows that this one single protein ZAR1 in Arabidopsis is able to recognize up to 40% of the Pseudomonas syringae pathogens on the planet. So it's really become remarkable receptor. So we just recently we asked whether this could be useful in our agriculture. So we are looking at here is the Chinese gooseberry. Basically, is Bihutao. You know, all kinds of different varieties. You know, it's very, very beautiful fruit. What's important about this fruit that this is important crop for farmers in the poverty-stricken areas in China. So this is called a big fruiting program. Okay, but in the last 15 years or so, a disease called bacterial canker result, which turned out to be a devastating disease for the food industry in the entire world. So once the orchards is infected, you have to chop down all the trees. So this is really bad. So a student in my lab recently looked into whether ZAR1 could potentially provide us a solution for this bacterial canker disease. She identified that in Arabidopsis that ZAR1 actually can recognize one effector protein called HAP-Z5 from this bacterial pathogen to cause resistance. Here's in Nicotiana bassiniana also this ZAR1 can recognize this HAP-Z5. So we are very interested to follow this to see whether this could potentially provide a remedy for this devastating disease. So I think the time is up with the. I'd like to thank my collaborators and also the contributors to the work. So particularly, I'd like to thank Chen Yihang, He Kangmin, and also Cai Jijie, my long-time collaborator. And the people did the work, the major contributors, including Bi Guozhi, Hu Meijuan. Thank you all.
Okay, any questions? since you guys first time to hear the plant immunity, right? So doctor, so I have like a question. I don't know. It's so you know, so the ZAR1 complex which makes a hole on the in the membrane, and you know, so the leaking of the calcium or sodium or whatever. So what about the proteins such as like DAMs, which is really small peptides?
That is an outstanding question. So DAMs there are actually different kinds. So there are some of them which are small, like ATP or NAD, which can act as a extracellular similar molecule DAMs. Others are peptides or small proteins. Okay, this ZAR1 is really small. It's only so small that they only allow ions to pass. Okay, so it's like 6 to 7 angstrom in the interior diameter. So it's small. It does not allow passage of peptides. Okay. In plants, there are also some peptides appear to regulate immune function in a way analogous to animal cytokines.
Yeah. So actually, so the ZAR1 complex, which is mediates also the ion secretion or whatever. And what about the other, like which is a known plant, like ion channel proteins? Are they involved all in the purpose in this process, or they are not doing anything?
Well, this is again an outstanding question. So this is a very active area of research. So there have been quite a few canonical calcium channels reported in that seem to operate in the plant immune system, but not shown to act downstream of NAR proteins. Okay, so those calcium channels, including CNGCs or glutamate-like receptors, they act downstream of cell surface receptors. But I think that the jury is still out there whether they can function downstream of NAR protein. That's not concluded yet.
Okay. Dear Dr. Zhu, so thanks for the wonderful and inspiring talks. So my question is also about this channel. So there are, to my knowledge, there are two very unusual points about this channel. First one is it's voltage gated. So it's a voltage gated cation channel. So have you ever checked about this structure? Have you ever found out which part or which residue are responsible for the voltage sensing function? My second question is about this E11 residue. To my knowledge, it's also very unusual that when you mutate this glutamic acid to alanine, it will negatively regulate this channel. That's very unusual because it's a smaller side chain for alanine than glutamic acid, I think.
That's an outstanding question. So the first two questions. First is about what are the gating mechanisms, like voltage gating mechanisms. So at this point we don't know yet, but I don't think there's a gating mechanism. I think this is unusual channel that once formed, it hits a point of no return because it keeps fluxing in the calcium. It does not close. About the glutamate residue, there are actually several negative charged residues in the channel cavity, but we mutated all those, and they did not seem to affect the function. So the substitution of this glutamate with aniline, I think the key point is not about the side chain, is about the charge. I think without the charge, without the negative charge, the side chain, then the cation is not selected, is not allowed to pass through. I think that this is in a way like the channel selection filters in other known channels. But nonetheless, this is an unusual or a non-canonical channel. Okay, is receptor turned into channel.
Check about one thing. There are only 5 transmembrane helices in this channel, right? 5, only 5.
Yeah, only 5, that's right, only 5.
So it seems that it's not this 5 transformer helices responsible for this voltage sensing, I think. So maybe the subsolic part. Oh, thanks for your talk. Yeah.
Thank you.
Any questions? Okay, if we don't have any questions, so please join me to thank Dr. Zhou and as well last 2 speakers, and we will come back at 3:30, I guess. Okay, have a break. Thank you.
Thank you.